One second, just let me plug in a few things and then we should be ready to go. All right. All right, so hi, this week is going well. We are going to go over the next two lectures. Happy Friday, good morning. We're going to go over the next two lectures for neuroscience. So we have the first lecture, which will be on neurochemical circuitry, and then we'll talk about the autonomic nervous system on the last one, right? So does anybody have any questions before we begin?
Real quick, out of the like 200 professors we have for this class, who writes the hardest questions, or do you even know on the exam?
So it's hard to tell because it's a lot of it's like mixed in between, right? So a lot of the questions are going to be you don't know which professor is going to be from because it doesn't say like the specific name, right? So I would say it's just going to be a mixture of typically the neuroscience exam is, I would say the first one is pretty difficult. So I would say most of the professors definitely write tough questions. But this one, I would say the first one that we're going to do right now, she's pretty straightforward, Dr. Blair. So I'll definitely highlight the things that are high yield for her because she's really straightforward when it comes to her questions.
Yeah, she was pretty chill. All right,
Yeah,
thanks.
Yeah, I got you. All right. Any other questions? All right. Let me just pull up the lecture real quick. Yeah, there's so many professors in the course. They change all the time. Okay. All right, can everybody see my screen? We're good to go? All right, perfect.
Yeah.
All right, sounds good. All right, so we're gonna go over neurochemical circuitry. So basically, I'm definitely gonna one second. I'm definitely going to talk about the high yield topics that are going to be important for the exam because she's really straightforward when she talks about the PowerPoint. So I made sure to make a few slides here and there that are basically kind of like summary slides, I would say, or basically all in one that would make sure that would help you make sure to highlight the highest of yields, the things that she pointed out that she wanted you to know for the exam. Because there's a lot of stuff in this lecture that I would say good information to know, but it's most likely not going to be tested on because she is really straightforward when she talked about this lecture, and I'm definitely going to highlight on those things. So you'll see what I'm talking about when I go throughout the lecture. All right, these are the learning objectives. So we'll be talking about some neurotransmitters, neuroanatomical pathways, and then agonists and antagonists at the end of the lecture. Okay, so I feel like a lot of us know this. Remember that summation occurs for all neurons. That's the main purpose of communication, right? It's not just one-to-one. It's a summation of signals, a summation of inhibitory or excitatory. Remember, we talked about that a lot with Dr. D'Agostino's lectures. So this slide should be a review. And then we have single neurons. Remember, they're just complex, right? So dendrites, axons, myelin sheaths, so many synapses, et cetera, et cetera. Just another slide basically reinforcing the previous one. Okay, so for synaptic transmission, remember again, we have chemical versus electrical. Remember, chemical is much slower, right? So again, that's our process of producing the neurotransmitter, putting it in a vesicle, moving the vesicle to the presynaptic terminal, and then releasing it into the synaptic cleft, and eventually binding to receptors, and then opening those ion channels, and then influxing sodium, etc. Right? So that's such a long process, which is why chemical synapses are much slower than electrical. You can see these electrical synapses are just connected via gap junctions, which is why they have smaller synaptic clefts. They shoot signals basically down these neurons, so they're much, much faster. Again, just another review. Remember, calcium dependence is very important. Remember, when calcium influxes into the neuron, that's very important for synaptotagmin, which is the calcium sensor. Remember that, it's really important. Synaptotagmin, which is the calcium sensor, which is going to help move the vesicle to form that core complex, which includes snares and snaps. And that's eventually going to cause the efflux of, let's just say, acetylcholine, right? So it's very calcium dependent regarding these chemical synapses. Okay, so these are just some of the classes of neurotransmitters. We'll touch upon some of these, but not all of them, right? So acetylcholine, right? We'll talk about that. GABA, glutamate, etc., as well as the unconventional. Unconventional are really interesting, so we'll talk about those when we get there. All right, so again, this is the same thing we just talked about, just a life cycle of a conventional neurotransmitter. What's really good about this PowerPoint is that the first few slides are going to be a review, right? So stuff we've already talked about with Dr. D'Agostino. So again, this is just the action potential coming in, influx of calcium, calcium mediated by synaptotagmin. Then it's going to move to the vesicle is going to move to the presynaptic terminal, release the neurotransmitter, and then eventually bind, right? So norepinephrine, dopamine, or for example, serotonin can be either inactivated or used to be reuptaken. So basically, it can be reuptaked. And then you have enzymatic hydrolysis, which is going to be through typically acetylcholine through acetylcholine esterase or histamine, right? All right, so the next few slides I basically made were kind of like summary slides. So I talk about like unconventional neurotransmitters, and I put like I would say 2 to 3 slides on this on what I feel was the highest yield. So I feel like I know people study in different ways. So I wanted to. So for me, I like things in bullet points, like straight to the point. So I made those slides like that. So because I know you can study off the slide as well, but again, I put the highest of yields in these so that you have it all in one spot. Right, so unconventional neurotransmitters, the first thing we should start thinking about are obviously things that are not, conventional neurotransmitters, right? So like carbon monoxide and nitric oxide, right? Nitric oxide is a very potent vasodilator, right? Because it goes through nitric oxide synthase. It's also very associated with regulating with cyclic GMP, which is also helping with vasodilation. Carbon monoxide, it's actually going to be produced by heme oxygenase. So these are going to be important to note. So the enzymes, I would say heme oxygenase and NOS, nitric oxide synthase, are going to be important to note because NOS is going to produce nitric oxide, while heme oxygenase typically is going to liberate carbon monoxide, right? And these can be basically neurotransmitters, but what's interesting about them is they're not present or stored in the vesicle. Right, so they're not stored; they're just made when these enzymatic reactions happen, right? So when heme oxygenase is like, let me just produce a carbon monoxide, right? That's not basically going to be stored anywhere. It's just going to be used quickly and spontaneously react to oxygen, right? So it's not released via exocytosis, of course, and it's too small to have any receptors, right? So carbon monoxide and nitric oxide don't have any receptors to bind to, right? So they usually just diffuse and is synthesized and quickly released on demand, right? So that makes sense. Again, through these enzymatic reactions, nitric oxide synthase or heme oxygenase, they're going to be producing these enzymes, or these enzymes are going to be producing these byproducts, and they're going to be very important for unconventional neurotransmitters. And to terminate them, carbon monoxide is going to spontaneously react with oxygen because we know carbon monoxide obviously can be toxic, right, at high doses. And then nitric oxide, which is a potent vasodilator, is going to spontaneously react with oxygen as well, right? So those are the key points in regards to carbon monoxide as well as nitric oxide, right? So I would say these are the stuff that she can definitely test on for the exam. So I would highlight these points and the enzymes as well. Then we have BDNF and NGF. These are actually really interesting unconventional neurotransmitters. They belong to the neurotropin family. So basically what slides are those? Gabrielle, what are you specifically referring to? Are you talking about the carbon monoxide and nitric oxide?
Yeah, I just don't understand what those two slides you just went over. I don't have those on my
Oh,
PowerPoint. I was trying to follow along. I don't.
yeah. So I said that like I made these slides because I wanted to put bullet points basically of high yield stuff that would for sure the things that she would test on. So these are just slides that I made basically.
Okay, so we don't have these slides then.
No. Yeah, I mean you will obviously right when I post it. Does that make sense?
Yeah.
Okay, for sure. All right, so BDNF and NGF, this is so basically if you wanted to follow, you would just obviously skip through the slides that talk about carbon monoxide, nitric oxide, BDNF, NGF, right? So again, she obviously has slides on that, but I wanted to make summary slides that are very helpful in regards to what is important, straight to the point, bullet points. So BDNF and NGF, they're actually really interesting. The first thing you should start thinking about with BDNF is that it's very important for synaptic connections, neuronal survival, as well as neurogenesis, right? So BDNF there's lots of studies going on BDNF and NGF, and they're very important for like increasing connections in the brain, very important for plasticity. So I put this little picture here. You don't need to obviously know this stuff, but it's super interesting knowing that like exercise can increase your BDNF. Less sleep can decrease your BDNF, and BDNF and NGF are very important for obviously plasticity, connecting neurons, neuronal survival, very good things, right? So for example, in Alzheimer's disease and stuff like that, you have low levels of BDNF and NGF, right? So less connections, less neuronal survival. So BDNF is actually a very important neurotransmitter physiologically, right? So it's constitutively synthesized and released, right? So from neuronal depolarization, depending if it's on the soma, on the dendrites, or the nerve terminals. And again, remember, it's very important for neurogenesis, neuronal survival, and excitability and connectivity, right? So that's why I put this on here. And this is just the last one just to talk about BDNF. So basically, she stated that it's synthesized in the endoplasmic reticulum, right? So it's an endoplasmic reticulum produced peptide, right? So too much of BDNF can actually affect your plasticity, right? So obviously you don't want too much of anything, everything in moderation, right? That's the quote, right? It's typically good. So it affects plasticity and may affect learning. So if you have too little BDNF, we know that can lead to decreases in plasticity and increased effects of depressive episodes, right? Because again, you don't want too low levels of BDNF, right? So no need to study this image. It's just to show you how like BDNF is just like every other neurotransmitter, right? It's produced in the endoplasmic reticulum, goes in the vesicle, broken down by proteases because it's a peptide, right? So it goes from its pre-pro to its pro. And eventually BDNF comes out and binds to these neurotransmitters. You don't need to know these. She said that you don't need to know these, and then eventually indicate a response that helps with neuronal survival and synaptic plasticity. Right, so I added this picture just to show you that these unconventional neurotransmitters, some of them just do the same exact things that normal neurotransmitters do, right? So it's really interesting to see this work the same way. So basically, those are the summary slides, right? So I wanted to add a little quick question in there, and then we'll move on. So which of the following is incorrect regarding carbon monoxide and nitric oxide signaling, right? So I'll give you a few minutes, and if anybody has any questions, feel free to let me know.
Hey Justin, this may be a stupid question, but I think I'm blanking this morning. So all neurotransmitters are kind of made in the
So they could be, right? So not all of them. So for example, like like acetylcholine, which is
yeah, like the
acetyl-CoA plus.
peptide ones.
Yeah, I would say typically the peptide ones, yeah, would be.
Okay, sorry, I don't know why I'm like forgetting.
No, you're good. No, you're good. You're good. You're good. It's Friday. Y'all good. It's all good. Okay. So, yes. All right. So, which of the following is incorrect regarding carbon monoxide and nitric oxide signaling? Everybody seems like they're on the same page, which is really good, right? So it's basically going to everything is correct except for B, right? So signaling cascade via receptors. Remember, carbon monoxide and nitric oxide, they're very small molecules. Typically, they just rapidly diffuse through the cell, right? So they don't need any receptors, and they spontaneously react to oxygen, which is good, right? Synthesized and released on demand, and not present or stored in vesicles, right? So really good job on that one. So basically those summary slides that I made were basically on these slides that she talked about, right? So I'm just going to kind of like skip through these slides or basically highlight a few things. But again, that's why I made those summary slides, because she's really straightforward. There's a lot of things on the slides that typically won't be high yield for the exam. So which is why I highlighted those summary slides, right? So this is exactly what we talked about, right? We talked about nitric oxide also being very important for cyclic GMP, right? So one of the important things here is also knowing that arginine, right? Arginine is going to be the amino acid that produces nitric oxide. But again, I have that in the image on the summary slide. So it shows you that arginine via nitric oxide synthase is going to produce nitric oxide, right? So that's going to be very important to also note as well, is going to be the amino acid that helps produce nitric oxide. And then again, this is carbon monoxide. So we showed the enzyme, remember, in the first summary with heme oxygenase, right? So heme oxygenase helping to produce basically carbon monoxide as a byproduct, right? And then a carbon monoxide regulates cyclic GMP and spontaneously reacts with oxygen, right? And then this is our BDNF. We highlighted this, a very, very cool molecule, right? Unconventional neurotransmitter, right? Always released and always synthesized. One of these peptide neurotransmitters right here, and it's going to be, remember, again, important for neuronal survival, right? When you think BDNF and NGF, start thinking like neuronal survival, connections, etc. Very good things typically, right? And then again, you don't need to know the receptors, right? She said not to worry about them. I showed a picture of it just to show how it works, but again, you don't need to know that. And again, these are the indicated effects, as well as what we talked about regarding too much and too little. Right, so too much BDNF can obviously affect plasticity. Too little would lead to, for example, a depressive episode or increased chances of, and also decreased plasticity, right? So again, this is why I made these summary slides, because I like them straight in bullet points, and I know people study in different ways. So again, you can study straight off these as well. But again, I made those summary slides as well to help you out. Okay. So neurotransmitters, so we have our small molecules, right? So we'll talk a lot about GABA, glutamate, or actually almost all of these basically, and we're going to start highlighting them as we move on. Okay, so basically, so modulating neurotransmission, so whenever people say an agonist to this receptor, right, this is an agonist, right? For example, nicotine is an agonist to nicotinic acetylcholine receptors, right? So they can help activate the receptor. Antagonists basically block the agonist from binding, right? So anything that's going to basically oppose the action of the agonist is considered an antagonist, right? So it'd be the opposite effect, right? So this comes into play a lot basically in pharm, right? For example, we have a lot of agonistic drugs as well as antagonistic drugs, which will just do the same or opposing effects regarding the receptors that they influence, right? So again, those are going to be the main differences between agonist and antagonist. All right, so another summary slide here, really quick. Again, just highlighting the key points. If you want to obviously follow along, just follow the slides. Let's talk about acetylcholine, right? So again, the importance of acetylcholine, we know it's a very important neurotransmitter in the body. It's very important for muscle contraction, right? So at the synaptic cleft, so very important for muscle contraction. It's also very important for other movements, which we'll talk about in the sympathetic and parasympathetic. So important here, choline and acetylcholine produce acetylcholine. Remember, acetylcholine comes from the mitochondria, right? And this is via acetyltransferase, which is CHAT, right? So basically, choline acetyltransferase, that's the enzyme. And again, this is very high yield, knowing that the rate limiting step of acetylcholine production is choline uptake, right? So because typically, obviously, you're going to have a lot of acetylcholine usually because that's running through your Krebs cycle, right? So that's going to be always constantly going. So you're typically going to have a lot of acetylcholine or enough basically to make acetylcholine. So choline uptake is definitely going to be the rate limiting step of this production, right? So that's important to know. Acetylcholine esterase, so we know it's going to terminate acetylcholine, right? So it's going to break it down in the synaptic cleft. Remember, in myasthenia gravis, we use inhibitors of acetylcholine esterase so that you can increase the number of acetylcholine in the synaptic cleft, right? We talked about that in D'Agostino's lecture. And then we have our nicotinic and our muscarinic acetylcholine receptors, right? Nicotinic being ionotropic, and then muscarinic being metabotrophic, right? And then affects muscle movement, neuronal plasticity, memory. So you can assume basically if you just shoot basically or inject acetylcholine into your neuromuscular junctions, it can lead to serious muscle contractions, right? Because too much acetylcholine can lead to increased muscle contractions, spasms, etc. Right? So too little acetylcholine can lead to paralysis, right? So if you don't have enough acetylcholine binding to those receptors, you won't be able to send action potentials, which can lead to paralysis, right?
That's another question.
Yeah, of course.
So with the acetylcholine and its effect of having too much or too little, we should learn this completely separate from her summary slide at the end with function and dysfunction of the pathways, because those are specifically talking about the function or dysfunction of the pathway that is affected by this neurotransmitter, correct?
Yeah, very good point. Yeah, so this is the only one I think that, or one or two of them that I don't put the dysfunction and functions. So in the other summary slides, I do put dysfunction and functions so that you can separate that between too much and too little.
Okay. All right. Thanks, man. I was just wondering about that.
Yeah, gotcha. That's a really good point. Yeah, for sure. That summary slide is also going to be very important. We'll talk about that at the end.
Cool.
Got you. All right. So basically, again, that's just basically highlighting what we talked about here. So typically, if she doesn't underline it, right, it's not going to be very important. Typically, it's like not going to be important at all, right? So it's cool to know that, like, obviously you have a vesicle transporter that's going to move acetylcholine into the vesicle, right, driven by a hydrogen gradient. That's actually super important. Super important, not for the exam, but like outside knowledge. And then obviously hydrolysis through acetylcholine esterase. Again, this is what we talked about, right? So I basically tried to put 3 slides or 4 slides into one slide so you can summarize it into what's really the most important things to note. All right, so cholinergic circuits, right? So basically, this is what she's talking about now. So cholinergic, so when you hear cholinergic, the first thing that should be in your mind is acetylcholine, right? So acetylcholine, cholinergic. They mainly, of course, produce acetylcholine. They can produce other neurons, but typically they're mainly going to produce acetylcholine, right? So the locations of cholinergic neurons are going to be in the basal forebrain, right? So it's going to be located in the basal forebrain. You have that's where you typically have your nucleus accumbens, your medial septal nucleus, right? So that's going to be that area right here in that circle right here. And it's important for cortical activation, right? So basically of the cortex, cognitive function, and memory processing, right? So you can assume basically if there's effects basically or a dysfunction basically in these cholinergic neurons in the basal forebrain, it can lead to it actually as caused by degeneration and Alzheimer's disease, right? Because you won't be able to have these memory processings, these cognitive functions, and cortical activation. So there's actually decreased levels of acetylcholine, or cholinergic neuron production, right? And then you also have them in the brainstem neurons, right? And it also induces REM sleep, right? So these are two main locations basically that you want to know that are going to be where the cholinergic neurons are going to be produced, right? So this is going to be important to know. So here's the basal forebrain, and this is the brainstem. All right, so BDNF is synthesized in which location, and too little of it can lead to which scenario?
Hey Justin, this is probably a dumb question, but when they say circuits, do they mean like life cycles of the neurotransmitter, or like is there a synonym for circuits?
Yeah, so when they say circuits, they just mean like areas, for example, in the brain where neurons, this specific neurons that target basically producing only acetylcholine or other basically neurotransmitters. So basically this cholinergic circuit, basically where it's basically there's a lot of neurons that are producing mainly acetylcholine, for example, are located in the basal forebrain and brainstem. Does that make sense?
So it's like where they're produced, just mainly?
Mm-hmm.
Okay, thank
Yep.
you.
Yep. All right, so BDNF is synthesized in which location, and too little of it can lead to which scenario. So we have endoplasmic reticulum and depressive episode, right? So again, remember, first thing that should come to your mind when you hear BDNF or NGF is neuronal survival, neuronal connections, neurogenesis, etc. Right? So very good job with that one, right? All right, and that's basically what we highlighted. Basically, were these cholinergic circuits, right? So you see here in the basal forebrain, right, just like we talked about, as well as the brainstem, right? So from the basal forebrain, it can go to lots of locations, right? You don't need to memorize these, right? So it goes to the cortex, amygdala, et cetera, et cetera. We talked about important for cortical activation, memory, and it also could be degenerate in Alzheimer's disease, right? So again, these cholinergic circuits, or basically where these acetylcholine producing neurons mostly are located, which is the basal forebrain and brainstem, can be affected. Right, so again, brainstem neurons are usually important for activation inducing REM, right? So I highlighted those things on the slide previously. This was her question that she asked, so in the lecture, so I feel like a lot of us have already done this. This was going to be C, right? So acetylcholine neurons degenerate in Alzheimer's disease. How would you target this pathway? Acetylcholine esterase inhibitors. Okay, she already went through that in the lecture. All right, so I'm trying to maintain basically acetylcholine signaling in Alzheimer's disease to preserve cognition, right? So again, remember what we talked about in the basal forebrain, right? So the two main areas we have acetylcholine production is the basal forebrain and our brainstem. And remember, in the basal forebrain, it typically when you have these decreased levels of acetylcholine, it's shown in Alzheimer's disease, right? So sometimes in Alzheimer's disease patients, they can use acetylcholine esterase inhibitors in order to increase the acetylcholine and hopefully improve cognitive function, memory, etc. Right? So that's why there's some benefits into using acetylcholine esterase inhibitors for Alzheimer's disease to increase the number of acetylcholine at the synaptic cleft, right? All right, so this is going to be another summary slide. So again, if you want to follow along, just follow the serotonergic circuit. So circuit, again, this is just going to be where neurons in the brain produce mostly serotonin, right? So this is going to be mostly in the raphe nuclei. So you can see this right here. This is in the brainstem, right? This is the raphe nuclei in the brainstem. And that's going to be the area where it's very important for serotonin production, right? So a lot of serotonin is produced in that raphe nuclei, right? So that's very important to know, right? So produces serotonin. Again, an important amino acid that produces serotonin is going to be tryptophan, right? So remember, tryptophan produces serotonin. So again, we got to get these amino acids knocked down because a lot of amino acids that are producing different structures, right? Tryptophan producing serotonin, right? So that's going to be one of them. We have arginine producing nitric oxide, right? So different amino acids producing different structures, right? And this is via tryptophan hydroxylase. So serotonin can be reuptaked by the presynaptic terminal and catabolized by MAOA, which is just monoamine oxidase, right? So typically, monoamine oxidases are going to break down serotonin norepinephrine, dopamine, etc. Right? So we highlighted this in Dr. D'Agostino's lecture, but remember that the only serotonin receptor that's ionotropic is our 5-HT3. All other serotonergic receptors are going to be GPCRs, right? So here, basically, I'm highlighting basically what the summary slide talks about, which is the effects, right, and the dysfunction, right? So again, we know serotonin is important for like mood control, right? It's also important for circadian rhythms and homeostasis, right? That's the main effects of serotonin. Dysfunction of serotonin can actually lead to schizophrenia as well as depression, right? So you have dysfunction in these circuits, or dysfunction in the production of the serotonin can lead to these serious diagnoses, right? And then we have SSRIs, which are going to be our serotonin reuptake inhibitors, right? So if you inhibit the reuptake of serotonin, that means there's more serotonin in the synaptic cleft, right? So they typically try to use that to treat depression, panic attacks, right? So that's why they're blocking the reuptake, right? And then here we highlight our too much serotonin, which is obviously not good. That can lead to serotonin syndrome, which is confusion, twitching, trembling, right? Because again, we know serotonin is typically associated with like happiness, right? So, well, I mean, somewhat happiness, right? Not always, but somewhat happiness. And then obviously, if you were to increase serotonin too much, it would lead to twitching and trembling, right? But too little of it can lead to sleep changes, mental health problems, etc. Right? So again, you can see this is our raphe nuclei. We have like a rostral and a caudal, right? So the rostral, which means like closer to the top of the head, caudal means like closer to the tail, right? So these are like our rostral raphe nuclei, which is up here, right? They project everywhere, right? They can project to the cortex to send serotonin. They can project to the limbic system, the hypothalamus, right? So all these areas, our caudal right here, which is our raphe nuclei, is more so, I would say, important with like motor and like non-motor function, right? So a connection to the spinal cord because it's much lower, right? So basically, raphe nuclei is important for serotonergic production. So basically, I'm just gonna skip these, right? So this is exactly what we talked about. Again, highlighting. So again, see, there's a lot of effects here, but I wanted to just highlight the key ones that are important, right? So you don't need to know all of them, right? She just wants you to know the very important effects, which is why I highlighted those. Too much, again, can lead to these, but you don't need to know confusion, twitching, dilated pupils, shivering, goosebumps, headache, right? That's just, I would say, too much. That's why I just wrote confusion and twitching as the most high yield, and then mental health symptoms and sleep changes, right?
I have a question.
Yeah, of course.
So will she, like when she's asking a question based on the effects, will it be more of like, will it like she ask like clinical type questions, or say like, oh, the person is experiencing this and these types of symptoms, like what, what is it,
Yeah, I mean, she can ask a little bit of both, right? So she can ask in that sense, and she can also just straight up ask which area is important for serotonergic circuits, right? And you just have to know it's raphe nuclei, right? Does that make sense? But
Okay,
typically, I
well,
would
yeah,
say she's more so straightforward.
Oh, okay, yeah. I was just wondering because how important like the effects are, like when it's high or low, like.
Oh,
Yeah,
that's important. Yeah. So you want to know. So basically, what I put in the summary slide, you definitely want to know like what's happens high or low, because you can obviously test in that way. If you have too much serotonin, what can happen? Like she can ask if you block a if you block a serotonin reuptake inhibitor, what's gonna happen, right? So those types of things. So you want to know what too much of serotonin can cause, or what too little can cause as well. Does that make sense?
Yeah, okay, thank you.
Yeah, got you. All right, and this is what we talked about on the previous slide, right? And then don't know the drug examples. You don't need to know that. So don't worry about that. That's for pharm, right? I don't even think you touch on all these for pharm. So, and then depression and panic attacks, we talked about. That's the treatment that you use SSRIs for. All right, so you see, like I tried to make like 3 or 4 slides into one, because there's really is a lot of things that are just like obviously good facts to know, but a lot of the things that are testable are going to be on these study slides, right? So I would say if you study these study slides and you study the summaries at the end, you should be very, very, very prepared for this section of the exam. Okay, so dopaminergic circuit, remember that's going to be where you produce dopamine, right? So where lots of neurons are going to produce dopamine. So that's going to be tyrosine. So another amino acid that we need to know, right? Tyrosine is going to produce dopamine. And that the rate limiting step is going to be tyrosine hydroxylase, right? We talked about this in our previous lectures, but remember again, tyrosine hydroxylase is the rate limiting step of producing dopamine, right? And also remember choline uptake is the rate limiting step of acetylcholine synthesis, right? So there's a lot of rate limiting steps that we want to know. Reuptake to presynaptic, and then all dopamine receptors are GPCRs, also known as metabotropic, right? So key thing to know, dopamine, basal ganglia. So the first thing when you hear basal ganglia, the first thing that should come to your mind is like dopamine, effects in Parkinson's disease, movement, right? That's what the basal ganglia is highly associated with, right? So you have your ventral tegmental area, which is actually, it's studied a lot basically, which is important for goal-oriented movement. It's also important for reward motivation. So typically the ventral tegmental area is important for motivation. So that's the area that's usually targeted in drug abuse as well, because that reward motivation actually is going to be stimulated in the VTA, right? So that's why drug abuse is typically associated with the VTA. Substantia nigra, there's actually two parts of the substantia nigra, which we'll learn about later, but specifically the pars compacta is going to be important for posture and movement, right? So you have basically the substantia nigra, is like it shows in the image right here. It's important for producing a lot of dopamine, right? So it actually has a very dark stain, so meaning that you have your dopaminergic neurons are working. But in, for example, Parkinson's disease, that dark area is gone basically. So basically meaning the dopaminergic neurons are dying, right? So you're not able to produce this dopamine, and dopamine is actually very important for controlling and facilitating movement, right? So it's degenerate in Parkinson's disease, right? So I would associate substantia nigra with Parkinson's disease if it's obviously low levels of dopamine. And ventral tegmental area associated more so with drug abuse, right? So if you have too much dopamine, we know that can lead to schizophrenia, and if you have too little dopamine, that can lead to Parkinson's disease, right? So that's why treating Parkinson's disease is obviously there's other factors that make it very difficult to treat, but obviously leading to too much production of dopamine can actually turn the switch to schizophrenia, right? So you don't want to have obviously too much dopamine, and you definitely don't want to have too little dopamine, right? So that perfect balance is going to be key. Yeah. I was just about to say that, yeah. So basically, so what's interesting about the dopaminergic pathway is that, so for example, if you were to, let's just say in Parkinson's disease, right? So the substantia nigra is very affected, right? So that pathway basically for the substantia nigra, which is typically going to be mesostriatal, right? When you say mesostriatal, that just means like from the mesolimbic system to the striatum. The striatum is like your basal ganglia and, well, the basal ganglia area, which is very associated with movement, right? So if you sever basically that mesostriatal area, obviously you can lead to Parkinson's disease, right? Because you're not able to control your movement, but you still can have your reward motivation via the VTA pathway, right? So that's why there's two different pathways for dopamine. So you can have your reward motivation intact and your movement not, or vice versa. You can have your movement intact and your reward motivation not, right? Because if the VTA is affected by, for example, drug abuse, right? So those are going to be obviously pretty cool things to note, right? But this on these slides, this is what's going to be really important. You can also note obviously the mesostriatal pathway if you wanted to as well.
Hey, Justin, so would she, like, on an answer choice, have mesostriatal pathway and substantia nigra, or just substantia nigra as an option for causing, like, Parkinson's disease deficit?
Yeah, so I would say it's definitely going to be substantia nigra, right? So I would say mesostriatal and like the pathway to the cortex is definitely going to be on the lower yield side, right? So obviously it's good to note, because when one affects, it doesn't mean it's going to affect the other, but she would definitely go towards substantia nigra or VTA for drug abuse. Make sense?
Yes, perfect sense. Thank you.
Okay, gotcha. All right, so that's the important things basically to highlight from those slides, right? So the straightforward things that are definitely going to be high yield for the exam. So again, that's just what we talked about here, right? So I'm just going to skip this slide, right? Vesicle transporter is important to note, but she doesn't underline it, right? So it's not going to be typically important for the exam. Here's our dopamine. No need to know like D1 like and D2 like. You're gonna get actually that later on when we talk about the pathways in the basal ganglia, right? So we'll talk about the D1 like and the D2 like, but no need to know that. I put the high yield effects, and then this is our too much and our too little. Here's our VTA as well as substantia nigra, right? So she talks a little bit about the mesolimbocortical pathway, which is this pathway of the VTA, right? And then you have your mesostriatal, which is going to like the striatum of the basal ganglia, right? And that's just going to be important for more so movement, right? So there's two different pathways here that can be important for dopamine. So which of the following locations is targeted mainly due to drug abuse, right? So we already talked about this a lot. So this should be pretty straightforward, right? Any questions? I'll give you a few seconds. Okay, so everybody seems like they have the answer choices in, which is good. Which of the following locations is targeted mainly due to drug abuse? And that's going to be our VTA, right? Ventral tegmental area. Remember, raphe nuclei, important for serotonergic production. Basal forebrain, along with the brainstem, is important for acetylcholine production. Substantia nigra, that's for dopaminergic, but specifically typically targeted in Parkinson's disease, and anything affecting movement. Right? So, excuse me. Damn. Okay, so basically we have a motivation basically is dopamine dependent. So in this study, it's basically showing a rat that is either saline administered or cocaine administered. So basically it's just showing how a rat can press the lever, right? So it presses the lever. If it's cocaine, typically it's going to press the lever a lot of times, even like maybe hundreds, even thousands of times. To get cocaine, but again, it's only a regulated amount of cocaine that's going to be released, maybe once, twice every 30 minutes or hour, right? So when that happens, basically, it shows that basically that the physical dependence is not like an adequate way to explain drug addiction, right? So just because your body basically is like asking for it doesn't mean you typically like want it in this scenario, right? So that's why it's saying that like motivation is basically the dopamine dependent thing. So your dopaminergic neurons are typically going to be activated when you really want something. So for example, here, cocaine, right? So we talk about the differences between dopamine and serotonin because dopamine is more so associated with wanting, right? So you can see in this rat, it wants basically more cocaine, right? And that's basically what's driving this action, right? So serotonin is more so associated with liking, right? So an example that I use in the upcoming slides is like that she talks about, like you may like to exercise, right, which is like a serotonergic thing, but you may not want to, which is dopaminergic, right, which is your motivation, right? So with dopamine, you want to associate that with wanting to do something, while serotonin, you want to associate that with liking, right? So serotonin, liking, and dopamine is more so associated with the motivation to do something and wanting. Yeah. So yeah, it's basically, it's going to be the chemical dependence as well as the physical dependence, right? So obviously physical dependence will assist in that, but it's not the sole reason to explain drug addiction, which is why it would be chemical dependence as well as physical dependence.
Hey, Justin, I'm sorry. I guess I just don't understand how like the rat pulling the lever really shows like that whole wanting thing.
Yeah, so basically, so the rat basically is going to hit the lever way more basically to administer cocaine than it would if it was just saline, right? So for example, in like 30 minutes, it might hit it like 1,000 times for cocaine, but in saline, let's say it just hits it like 40 times or 50 times, right? Because it doesn't really want the saline, right? Like it wants the cocaine because that's dopamine driven as well as physical dependence. Does that make sense?
Oh, I get what you're saying. So like a physical, like for physical dependence, he wouldn't be like hitting the lever, I guess.
Yep. Mm-hmm.
Gotcha. Okay, thank you.
Yeah, no problem.
Hey,
All right.
Justin,
Yeah.
what do you mean exactly? Sorry if you already said this by physical dependence. How is, because chemical, like I understand what that means, but the physical part, I don't.
Yeah. So basically it's just like so for example, I use the physical dependence as more so pressing the lever, right? So the more times it's pressing the lever, like it's um basically more times it's dependent on that. Okay, so motivate. So basically the chemical is more so associated with dopamine, right? So dopamine basically meaning that you using it for the motivation to do something, right? So that's going to be basically more so associated with chemical, and then you also have associations with like the body just physically depending on it, right? So basically the body needing it basically for example, like you need to eat, right? So for example, that would be sometimes like physical dependence that your body needs it, right? So but dopamine is more so basically going to be our motivation to do it. Does that make sense? Like sometimes it's physical needs that you need to sustain your body to like survival, for example, and sometimes it's needs for motivation regarding like dopamine or chemical pathways. Does that make sense?
Yes, it does make sense. I just wanted to make sure I was differentiating.
No, you're good. Okay, so yes, exactly. Yeah, dopamine is gonna make you want the drug. Yep. Okay, so again, like we said, remember serotonin associated with liking, dopamine associated with wanting, right? So again, you may like to be in shape, but you may not want to exercise regularly, right? So that's our difference between our serotonin versus our dopamine. And she talked about this in lecture. Okay, so we have noradrenergic circuits. So we have again, so tyrosine. Remember, tyrosine is going to be important for our catecholamines, right? So dopamine, norepinephrine, and epinephrine are all going to be produced by the amino acid tyrosine, right? So the rate limiting step again is tyrosine hydroxylase. Norepinephrine is the only neurotransmitter synthesized in the vesicle, right? So what happens is that you bring dopamine into the vesicle. Once dopamine is inside the vesicle, you actually have an enzyme that causes beta hydroxylation that forms basically this norepinephrine, right? So it's actually formed inside the vesicle. And just like most catecholamines, it's going to be broken down by MAOA, which just means monoamine oxidase A, and then COMT, which is for catecholamines. And then basically the most important thing to know for noradrenergic circuits is that they're produced in the locus ceruleus, which is going to be in this area of the brainstem. Right, and we know noradrenaline, right? So noradrenaline is going to be important for alertness, right? Arousal, homeostasis, even some movement, right? So we know that it's basically going to be typically noradrenaline is important for like excitatory stuff, right? You also have SNRIs, which are going to be serotonin norepinephrine reuptake inhibitors. These were found after SSRIs, right? And they're used to obviously also used to treat anxiety and depression, and that's going to increase norepinephrine and serotonin in the synapse by blocking reuptake, right? So we talked about SSRIs, which block serotonin reuptake. SNRIs block norepinephrine and serotonin reuptake in order to treat depression and anxiety. Right, so you would assume basically if you have too much norepinephrine, it would lead to anxiety, increased blood pressure. Too little is like lethargy and lack of focus, right? And this is going to be our locus ceruleus you see right here, having projections to, for example, the cerebellum, right, for movement, like we talked about, to the hypothalamus for homeostasis, to the cortex for alertness, arousal, etc. Right, so many areas. So this is what we talked about here. So I'm going to skip this one, right? We talked about this here as well. And then we talked about this here again. Remember, I told you like it goes to cortical projections, hypothalamic projections, limbic, cerebellar, etc. It just goes to a vast variety of places, right? So you just want to know locus ceruleus associated with noradrenergic circuits, right? We also have our lateral tegmental nuclei that's also associated with noradrenergic circuits, right? But more so, it's just locus ceruleus, right? And this is going to be to the brainstem and to the spinal cord, right? So whenever you associate noradrenergic, start thinking basically like rush, basically of I think of like rush of sympathetic responses, more so like anxiety, increased blood pressure, etc. right? So no need to know these drugs, right? So these SNRIs, you don't need to know these drugs. Just know SNRIs are used to treat depression or anxiety, and they block reuptake of serotonin and norepinephrine. That should be suffice, right? No need to know like the side effects, right, and stuff like that. Okay. So, any questions before I talk about glutamate? Oh, wait. Yeah. So basically the question in the chat, yeah, so we'll talk about that in the next lecture. MAOA is more so associated when you, for example, if you reuptake norepinephrine to the presynaptic cell, you have monoamine oxidases, which would break it down. But sometimes what can happen is that norepinephrine can actually go into the postsynaptic cell, right? And then it can be broken down by CompT, because CompT is more so associated with the postsynaptic cell. But again, it could be broken down by either or, right? Okay, so we have glutamatergic circuits. So again, where neurons typically produce glutamate, right? So remember that your neurotransmitter is going to be glutamine. So glutamine produces glutamate via glutaminase, right? Glutaminase is the rate limiting step. So again, you see I'm highlighting these in red, these rate limiting steps, because they're very high yield, right? And what's going on here is that, so you send glutamate for an excitatory response, right? So it goes to the presynaptic neuron, into the synaptic cleft, and then initiates a response. Right, but what happens is you don't want too much glutamate, because too much glutamate can lead to excitotoxicity, right? So which is why we have these astrocytes in these glia, which are in these synaptic clefts, which are very important for picking up these glutamate, right? And it picks up this glutamate via these excitatory amino acid transporter, right? It transports the glutamate from the synaptic cleft into the astrocytes, breaks it down into glutamine, and then glutamine is basically sent back into the presynaptic neuron, right? And then reused again to produce glutamate, right? So it's kind of like a cycle, right? So to make sure that this glutamate is not just freely out there promoting excitatory responses, right? It's very regulated via this enzyme, which is important to note, right? And then we have our glutamate receptors. We talked about those in Dr. D'Agostino's. It's widespread. So glutamate is actually produced not in a specific location. It's widespread throughout the central nervous system. Just like when we talk about GABA and glutamate are widespread throughout the central nervous system. Here are the main effects. Again, we talked about excitatory, learning memory, synaptic plasticity. And if you dysfunction of glutamate, that would lead to excitotoxicity, right? So we'll talk about in the last upcoming slides that glutamate has a central role in long-term potentiation, right? So I'll talk about that when we get to that slide. Right? And then lastly, too much glutamate leads to excitotoxicity, and too little can lead to obviously opposite effects, so psychosis, coma, or death. So this is our summary slide for glutamate, right? So the very high yield important things I would say that we definitely need to know for the exam. Right? And that's what she talks about here, right? So we went through this complex already. We went through this as well, right? You don't need to know muscarinic glutamate 1 through 8, right? So that's not too important. And then again, widespread throughout the central nervous system. We talk, it's an NMDA antagonist, right? Are neuroprotective, because if you go against NMDA, we know NMDA is important for glutamate, right? So it's a glutamate receptor. It actually can be used for a basis of Alzheimer's disease, Parkinson's, and may play a role in schizophrenia. Okay, so where's norepinephrine mainly produced, and what would occur if you had too much norepinephrine, right? I'll give you a few seconds.
Hey, Justin, while we were taking a minute,
Yeah.
What is the significance of, I guess, coupling the serotonin with the norepinephrine in the SNRIs?
Yeah, so I would say it's more so to increase the thought of it was probably to increase the effect, so to better treat basically maybe anxiety and depression, because you're allowing for not only more serotonin, more norepinephrine as well.
Okay, and so they don't really do just norepinephrine on its own, because serotonin's the main, I guess, liking or happy drug kind of, or happy neurotransmitter kind of thing. So it's
Mm-hmm. Exactly. Yep. Which is typically why you're gonna have serotonin as well.
Okay, so the norepinephrine enhance its effects almost.
Yep, exactly.
Got it. That makes sense. I guess I wasn't thinking about it from like the perspective of like what they're trying to treat. So
No,
thank
you're
you.
good. Yeah, you're good. Right. So obviously, so these have some mixed effects depending on the person, right? So it's more effective in some people than others, right? So it really just depends. All right. So where is norepinephrine mainly produced, and what would occur if you had too much norepinephrine? Remember, norepinephrine is our locus coeruleus, and too much would lead to, like everybody said, increased blood pressure. Right. Too little would lead to lethargy. Raphe nuclei, remember that's for serotonin, and then widespread is actually going to be for glutamate as well as GABA. But remember, glutamate is excitotoxicity. All right, so I think this is our last circuit, right? So GABAergic circuits. So GABAergic are going to be widespread, right? So produced throughout the entire central nervous system. Our rate limiting steps. So what's interesting about GABA is that it's made by glutamate, right? So it sounds like really counterintuitive, right? Like because like GABA is excitatory, glutamate's excitatory, and GABA is inhibitory, right? But that's going to be basically the amino acid that produces GABA, and that's via glutamic acid decarboxylase, right? So that's important to note. GAD is going to produce GABA via glutamate, right? So that's important. And then our termination basically of GABA is through a GABA transporter, right, into neurons and then catabolized basically by GABA transaminase, right? So basically it's just then broken down respectively, right? So just from Dr. D'Aristino, the only reason why I put this here is because I know it's important for Dr. D'Aristino's lectures. GABA A and C, remember, are ionotropic, and GABA B, there's a B in metabotropic, so GABA B is going to be metabotropic, right? So again, it's the main inhibitory neurotransmitter. When you think GABA, you always got to think inhibitory. So it's going to be effects anxiolytic, so against anxiety, right? Anticonvulsant, as well as regulating pain, right? And dysfunction, obviously, if you have a dysfunction of GABA, would lead to anxiety, seizures, just the opposite effects, right? So if you were to administer somebody too much GABA, you can oversedate them or over-relax them because it's inhibitory, right? Too little would lead to anxiety, right? So this is our summary slide right here. You can see GABA is extremely widespread throughout the central nervous system. That's what we talked about here, right? So, and then here as well, and here. Okay, so the clinical application here, we basically, so obviously no need to know the drugs, right? Obviously, if you want to know them on your own time, that's cool. But important to know is just muscle relaxants are GABA agonists, right? So think about it. If you're a GABA agonist, you're promoting that inhibition. So you're promoting that muscle relaxation, right? That's the most important thing to note here, right? So GABA agonist, muscle relaxation. And anesthesia. So for example, NMDA antagonist. So that means glutamate antagonist. The most important thing is going to be if you inhibit glutamate, I mean you inhibit excitatory effects, you would lead to anesthesic effects, right? As well as numbing or pain or sedation, right? So just opposite effects, right? So this, I would say, if you note this green box, you should be good to go. Nothing else to note here. And then here's our LTP. So we actually go into this pretty in depth, I think in block 2. So what happens is that glutamate is very important in long-term potentiation, which is important for memory. So what's happening here is that you have a lot of glutamate. Well, when you have glutamate, right, and it affects an AMPA receptor, right? Because we know it's a glutamate receptor. Sodium goes inside the cell, right? Sodium goes inside the cell because remember it's excitatory, right? It's going to cause a membrane depolarization. That membrane depolarization is actually going to trigger the release of this magnesium because you typically have magnesium ions that are blocking these NMDA receptors. So that's going to trigger the magnesium ion to be removed. When the magnesium ion is removed, that allows for activation of the NMDA receptor, right? And then calcium can then influx into the cell, leading to more of a depolarization, right? And then when this calcium goes inside the cell, we know calcium can affect a lot of metabolic machinery, right? It can affect a lot of things, gene expression, second messengers, etc. And it's actually going to lead to increased either synthesis of AMPA receptors or increased transport of AMPA receptors to the synaptic terminal. So you'll see when that happens, you have more AMPA receptors at the end of it, right? So when you have more AMPA receptors, that means you have more areas to receive information, meaning you have stronger connections and probably going to increase learning and memory. Right, so that's how glutamate basically is important for long-term potentiation, which is important for learning as well as memory. All right, so this chart very important. I put a ginormous star here, so I would know everything on this chart. We highlighted these in the summary slides, right? So again, I would go through how I would study this. I would go through the summary slides. I would go through these charts, which are going to be very important, and it will help you understand the highest of yields for the exam. And then same thing here. We went over all the functions and dysfunctions, structures and pathways, as well as neurotransmitters, right? So study this slide as well. And then this is not going to be tested on, right? But obviously, typically neurons have single transmitters, but you can release more than one type, right? So obviously, like we said, cholinergic neurons typically produce acetylcholine, but they can produce other neurons or neurotransmitters as well. And this is our summary. All right, any questions? All right. If not, we're gonna take a little break right now. So we'll come back at around 10:06, 10:07, right? So we'll take a little break.
GABA and
All right, let me just pull up this other lecture. Does anybody have any questions before we start? Okay, let me just share my screen. Okay, so all right, so here we're going to be talking about the autonomic nervous system. We'll talk a lot about the receptors, right? Muscarinic receptors, nicotinic, adrenergic, etc. And we'll definitely highlight the high yield topics as well, as to what is going to be important for the exam, right? So we'll go over this lecture, and obviously feel free to ask me questions whenever need be. Here's the objectives. All right, so this is just a little schematic showing the nervous system, right? We have our central versus our peripheral, right? Peripheral giving our autonomic and our somatic. Somatic is used for muscle movement, right? Autonomic is going to be involuntary, right? So sympathetic, parasympathetic. You also have your enteric, right? So enteric nervous system as well. All right, so we have autonomic and somatic nervous systems, right? So they differ based on their action, right? We know somatic is typically with skeletal muscle, so exercising, for example, or lifting weights, right? That would be somatic, right? It's going to be typically myelinated, doesn't form networks, and for example, if you were to damage the somatic nervous system, it would lead to paralysis and atrophy of muscles, right? Autonomic is more so associated with, for example, your gut, your heart, lungs, etc., right? Things you can't obviously voluntarily control, right? I mean, yeah, most things like heart rate you can't control, right? You also have the not myelinated, which makes sense, right? You don't need fast communication in the autonomic nervous system typically, right? So typically you don't need fast communication to your stomach, right, and stuff like that, right? That's not necessary. Somatic is more so I need to contract these muscles quickly, right? Which is why it makes more sense why they're myelinated, right? Forms networks, and spontaneous activity would occur if you have denervation. All right, so I made this summary slide, but this is basically talking about these 4 slides that basically show all the neurons, basically, or all the fibers, basically, of sympathetic versus parasympathetic. So basically, we have our sympathetic nervous system and our parasympathetic nervous system, right? Our sympathetic, remember, that's our fight, flight, or freeze. Yeah, why did I write peripheral? Okay, I'm going to change that. Yeah, I meant to write, obviously, parasympathetic. So parasympathetic nervous system, that's going to be our PNS. I'm just going to say PNS, SNS, so I don't have to write the whole thing. So typically, sympathetic is going to be associated with your thoracic and lumbar region, right? So we talked about this in anatomy last semester. Sympathetic is more so thoracic lumbar, or also known as thoracolumbar, right? And the ganglia are very close to the spinal cord, right? So your ganglia are of the sympathetic nervous system is very close to the spinal cord. So and eventually, basically, you have your preganglionic and your postganglionic, right? So what preganglionic means is basically, obviously, from the central nervous system, you send a signal out, it goes to a ganglia, and then from that ganglia, it affects other things, right? That's going to be preganglionic versus postganglionic, right? So preganglionic before the ganglia, postganglionic after the ganglia. So preganglionic for the autonomic nervous system is going to be cholinergic, and then postganglionic is going to be cholinergic, dopaminergic, or adrenergic, right? So it could be depending on whichever system, right? And they're typically going to have very long postsynaptic fibers, ganglionic fibers. So again, that makes sense because again, with a sympathetic nervous system, right? So for example, you're running away from something. Typically, when that response occurs, it's going to go from your central nervous system to your ganglia, right? The ganglia are typically close to the spinal cord for sympathetic, right? And then it's going to go basically to its effector organs, or effector muscles, depending on organs, etc. And that's why they're typically very long, right? Because going from the spinal cord, for example, all the way to my biceps, right, would be a very long fiber, right? And stuff like that. So basically, your sympathetic nervous system is going to be very long postganglionic. Your peripheral nervous system is typically going to be very long preganglionic, right? So he talks about this in the lecture, and what's really interesting is that parasympathetic nervous system is more so associated with organ specificity, right? So it's going from the body, basically, from the nervous system to the actual organ, right? The ganglia sometimes are actually on the organ itself, right? So sometimes basically for the peripheral nervous system, the ganglion are actually located in the organ. So you can see in this image right here on the right, you can see it going from the nervous system to the actual organ itself, right? That's that peripheral nervous system, right? And it's going to be basically preganglionic versus postganglionic, right? So with I gotta change that. With parasympathetic nervous system, it's going to be cholinergic all the way, right? So again, cholinergic for the preganglionic neurons and cholinergic for the postganglionic neurons, right? So again, very long preganglionic fibers for parasympathetic, very long postganglionic fibers for sympathetic, right? And cholinergic for everything except the postganglionic fibers of the sympathetic nervous system, right? So that's basically the highlighted things that are going on here. I would say the most important things that we definitely need to know in regards to the differences between the sympathetic and the parasympathetic nervous system, right? Any questions on this? Is anybody confused regarding this stuff, like preganglionic versus postganglionic, etc.?
Can you differentiate between the two, the pre and post?
Yeah, so anything before the ganglia would be preganglionic, right? So for example, from the central nervous system, for example, like the spinal cord to the ganglia would be preganglionic, and then which would be here, because this is the ganglion. And then anything after this yellow area, which would be ganglion, to the organs, etc., would be postganglionic, right? So over here, you can see the parasympathetic, it's going straight to the organs, right? You don't even see ganglion here because the ganglion are typically on the organs, right? So that's why we say the parasympathetic nervous system has very long preganglionic fibers, right? Because this right here would be the ganglia, and then it would give its postsynaptic fiber, right? So it has a very short postganglionic fiber, but very long preganglionic. Does that make sense?
Hi, Justin. I have a question.
Okay, never mind.
Oh, wait. Sorry, were you not finished? You can go ahead.
What were you saying? Does that make sense with the preganglionic versus postganglionic?
So is the pre, I'm confused. Is the pre going to the organ and then post? I don't understand how they're different things.
Yeah. Okay, so when you send a signal from the central nervous system basically to the ganglion, that's preganglionic, right? So basically I'm sending a signal to ganglia, which is then going to go from the ganglia to its effector organ, right? That's typically how it works, right? So for the sympathetic nervous system, you send a signal from the spinal cord to the ganglia. So for example, a random ganglion, right? So let's just say the ventral, actually you don't have a ventral, but whatever, dorsal root ganglia, that's typically sensory though. So you go to a ganglia, right? And then you go to the effector organ, right? So going from the ganglia to the effector organ would be postganglionic. Anything before the ganglion is going to be preganglionic. Does that make sense?
Yeah.
Are you sure?
I'm sure it'll make sense later. There's a lot of words going around.
I know, no,
There's
I
just
know.
like there's just like two parts to it. Like,
yeah, I know that they're two separate things. I was just asking for him to differentiate it, but I'll have to look at it later.
I got you. Yeah, no, for sure. So yeah, just one more quick overview. Just again, so you preganglionic. So you just send a signal from the spinal cord to a ganglia. In the sympathetic nervous system, the ganglia is typically close to the spinal cord, which is why this fiber is very short. This fiber going to the ganglia is very short. Then when you go from the ganglia to the effector organ, that's going to be postganglionic, right? And that's typically going to be very long for the sympathetic nervous system, and it's vice versa for the parasympathetic. Oh, right. And that's exactly what we talked about here, right? We broke it down here, right? You see our preganglionic. These are going to be preganglionic going to the ganglion, right? These are preganglionic right here. That's not, but this is going to the ganglion, right? And then you have postganglionic. So from the ganglion to the effector organs would be postganglionic, right? Same thing here, parasympathetic. You have preganglionic. Here's our ganglion over here. So anything after this is going to be postganglionic, which would be over here. This is why it's typically you have longer preganglionic fibers, right? Okay. So you have differences between branches of the autonomic nervous system and the somatic nervous system. You have sympathetic versus parasympathetic, right? So again, remember your sympathetic is going to be fight, flight, or freeze, and your parasympathetic is typically going to be with rest, digest, and repair. Hold on. 1 second. My Teams is acting up. What? Hold on. 1 second. Okay, sorry about that one. Hold on. Okay, here we go. Cool. Okay, so yeah, that's very important for us to know, right? So sympathetic versus parasympathetic, flight or fight and freeze, and then parasympathetic more so being rest or digest, right? All right, so this is what we talked about in the beginning. So typically the ganglia of the sympathetic nervous system are close to the blank and have long blank fibers, right? So this is again reinforcing the topic we talked about. Give you a few seconds as you answer that. So if anybody has any concerns, let me know, or any questions while we wait.
Hey Justin, I have a quick question.
Yeah.
So, the adrenal medulla, would that then be preganglionic or postganglionic?
Yeah, so in that image, it showed that it went straight to the adrenal medulla, right?
Yeah.
Yeah, so I mean, I think the adrenal medulla is a little bit different, right? So that would probably typically be an exception. Adrenal medulla is actually regulated by lots of things, right? So and releases lots of things that are important for sympathetic responses, right? So I would just associate adrenal medulla just knowing that that's just going to be innervated by sympathetic nervous system, and I think that should be suffice.
Okay, thank you.
Yeah, no problem.
Didn't for the adrenal medulla, he said something about like it being kind of like preganglionic in a way because then it sends epinephrine, which is going to be like your postganglionic neurotransmitter, but in the form of like a hormone. Is that what he was saying about that, or am I?
Yeah, no, yeah. So he was basically just saying that epinephrine gives you typically your broadest response, right? So your broadest response is sympathetic nervous system because it affects almost everything in the body, right? So adrenal medulla, heart, etc. Like the epinephrine is typically going to give you your broadest response, which is why he was like sympathetic is typically a broad effect. Parasympathetic is more so organ specific, like lungs, heart, GI tract, etc. Does that make sense?
Yeah, thank you. That's super interesting.
Yeah, no problem. Okay, so typically the ganglia of the sympathetic nervous system are close to the spinal cord, right? And they have very long postganglionic fibers, right? So again, postganglionic after the ganglion. Okay, so again, this is just going to be a quick summary slide regarding the sympathetic stimulation versus the parasympathetic stimulation, right? So again, this is the same exact thing basically of the chart that he has, right? So I just put it in bullet points or just put the highest yields in bullet points, right? So you know if you're, for example, excited or scared or etc., sympathetic stimulation is occurring. The eyes would dilate, right? Because you want to get more peripheral vision. That occurs by contracting this radial muscle, right? Because you can see these lines basically here are the radial muscle surrounding the eye. And when they contract, they pull the eye and open it, right? So they pull it and open it. So the radial muscle contraction is associated with dilation. Blood vessels, skin and splanchnic are going to constrict, right? Because you don't need blood going to your skin or to your GI tract when you're trying to run away or be excited, etc. Right? You need it more so to dilate in the skeletal muscle, right? So that you can have more blood to your skeletal muscle as well as your coronary, so like your coronary arteries, right? Lungs bronchodilate, so your lungs open up, so you're able to breathe, right? So you can run faster, run farther, etc. The GI walls are going to relax, right? Because you don't want to. The last thing you want is the last thing you want is constricting your GI while you're trying to run away, right? That's not obviously very effective, and that's why your sphincters constrict, right? So your sphincters, which would constrict, so that nothing obviously is going to be excreted or released. Right, same thing with the genital urinary, and then skin. So what's interesting about the skin is that you have, for your hair follicles, they actually have sympathetic innervation, and typically, you know how you get goosebumps, right? If you're scared, that's going to be a sympathetic response because your hairs that actually helps your hairs basically stand up. So I think that's pretty interesting, right? And sympathetic basically for sweating, right? So obviously you want to sweat when you're going through a sympathetic response, and this is basically your metabolism. Right, so this is basically just a little summary, but it makes sense if we just think it out, right? Sympathetic stimulation. If you basically think of what happens when you're excited, scared, etc., these things should align very well. And this is our parasympathetic stimulation, right? So again, what's important here also to know is that blood vessels, sweat glands, and hair follicles do not have any parasympathetic stimulation, right? So they have none, right? So hair follicles. So example, like I said, goosebumps, or you get goosebumps. There's no parasympathetic response to that, right? So you're not gonna get like not goosebumps, right? So there's no response to that. Sweat glands as well. And you also have blood vessels. These are the three things that don't get parasympathetic stimulation, right? Heart rate, decreased heart rate, contractility, lungs bronchoconstrict, and then the vice versa for the GI tract. Right, and that's basically the breakdown of this chart, right? So I basically made this chart basically into 2 slides so that you can break it down for you just to digest it a little bit easier, right? And again, what's important to note, remember that parasympathetic, you don't have basically any stimulation to the sweat glands, blood vessels, or hair follicles, right? So I would for sure know that. I would feel like definitely that's going to be important to know in regards to what is getting these types of innervations, right? And that's going to be the same thing we just talked about here. Okay, so dual innervation by the autonomic nervous system in an organ, right? You have mydriasis versus meiosis, right? Mydriasis just means dilation. Meiosis just means constriction of the eyes, right? So again, you have your radial muscle, which is going to do that dilation, right? So I told you this is going to be these lines. Affecting my cursor. Okay, so basically these lines basically are going to be radial muscle, so it's going to open it, basically cause dilation. Meiosis is going to be the ones that are surrounding the eye, that blue line, those blue lines, that's going to be the circular muscle, which helps make your eyes constrict, right? All right, so cholinergic synapses, right? So again, typically you're going to have a lot of cholinergic synapses in your cardiac, smooth muscle, glands, nerve terminals, etc. You have some sympathetic as well as somatic, right? So we know acetylcholine is very important, for example, for skeletal movement, which is why it's important for somatic. You also have, remember, acetylcholine is very important for parasympathetic response as well, right? With the muscarinic acetylcholine receptors, which we'll talk about in the upcoming slides. When you hear muscarinic, the first thing you should start thinking about is typically parasympathetic. And then you have some sympathetic basically regarding cholinergic as well. Okay. So basically this is how, again, we've talked about this already. This should be kind of a review, right? Remember our choline uptake is the rate limiting step, right? So choline plus acetylcholine via choline acetyltransferase is going to produce our acetylcholine, right? That's going to then be released at the synaptic cleft, right? Let's see what's going on here. And over here, an interesting fact what he said in lecture, I thought it was really cool that he was like acetylcholine esterase can break down 10,000 acetylcholine molecules in a second. I think that's just insane. That's just a lot of acetylcholine molecules that are being broken down. I think that's just crazy how efficient that is. And basically, it can interact with presynaptic and postsynaptic receptors, right? You also have some heteroreceptors, which are going to modulate basically transmission. You'll talk about these heteroreceptors actually in pharmacology. Right, these heteroreceptors are actually going to be very important in regulating basically when to release neurotransmitters. So basically, sometimes these heteroreceptors are going to tell your presynaptic neuron like, hey, release more acetylcholine, or hey, stop releasing acetylcholine, right? So these heteroreceptors are going to be very important when you talk about them in pharmacology eventually, right? Okay. So cholinergic receptors, right? So the first ones we're actually going to talk about are nicotinic, right? So we're pretty familiar with that. Remember, that's our ionotropic, right? They're very fast transmission, but they're short-lived. And then we'll talk about the muscarinic after. All right, so I would note these agonists, right? So you have nicotine, right? DMPP, as well as epibatamine, which is just going to be frog toxin, right? So those things are going to be typically nicotinic acetylcholine receptor agonists, right? So if they're agonists to that, they're going to induce basically depolarization, muscle contractions, at high doses, even spasms, right? So that's what these nicotinic agonists are going to do. All right, so if you were running away from an animal to get to safety, which of the following physiological responses would be appropriate? Give you all a few seconds. Does anybody have any questions while we wait?
Hey, Justin, just while we're waiting, we have 2 more lectures this week and then the exam, right?
I think so, yeah. It's coming up soon.
Have you all decided on the TA schedule for exam block?
No, we haven't yet, but I think my TA schedules are going to stay the same because histo's on Thursday, right? So, and I do my sessions on Friday. So I'm going to stick with Friday since it's after histo, and then neuro's on Monday. So I think my schedule is going to stay the same, but we'll let you know which ones are going to change.
Okay, thank you.
Yeah, no problem. Okay, so if you were running away from an animal, right, to get to safety, which of the following physiological responses would be appropriate, right? So everybody seems to be on the same page on this. So mydriasis, bronchodilation, and GI wall relaxation, right? So again, remember, mydriasis means dilation, meiosis means constriction, right? So that's going to be very important for our sympathetic response. Okay. So nicotinic acetylcholine receptors, we basically have them almost everywhere. There's a lot of nicotinic acetylcholine receptors. The first thing you usually think about is in the neuromuscular junction of skeletal muscle, right? So that's typically where you usually find them, right? You obviously have some in parasympathetic as well as in even sympathetic, right? So causing sympathetic responses in the kidneys, heart, smooth muscle, etc. Okay, so you have basically neuronal versus muscular, basically nicotinic acetylcholine receptors, right? So they're almost the same, right? Their subunits are slightly different. So so some obviously drugs or agonists can actually bind to nicotinic neuronal receptors instead of nicotinic muscarinic receptors, right? So they're slightly different, right? So the nicotinic muscle are going to be the ones that we highly associate with skeletal muscle contraction, neuromuscular junction, etc. And the nicotinic neuronal, right, is more so associated being in the ganglion, right? So they help with ganglionic transmission, right? So you usually have those NNs basically in the ganglion, just helping to pass from preganglionic to postganglionic, right? No need to know the subunits, right? You don't spend time studying the subunits. You don't need to. Okay, so we have our cholinergic receptors. You have your muscarinic, right? So muscarinic, remember, first thing that you should think about is parasympathetic. You're gonna have, and they're gonna be metabotropic, right? Because they're GPCRs. We have our cholinergic muscarinic agonist, right? So muscarine, pilocarpine, and oxytremerine, right? These, I would say, I would note the muscarinic agonist as well as the nicotinic agonist. You're able to separate them correctly, right? Obviously, muscarine is a muscarinic agonist and nicotine is a nicotinic agonist, right? But I would also note the other two that are going to be important as well because you can ask exam questions regarding muscarinic agonists, antagonists, etc., and you should be able to know what the effects are going to cause, right? Okay. So muscarinic receptors, remember, so whenever you have a drug basically that's used on muscarinic receptors, it's always going to be affecting parasympathetic response, right? So you have some muscarinic receptors in the sympathetic, but again, mainly assist just muscarinic receptors with parasympathetic, right? So parasympathetic, muscarinic. So here basically we have our muscarinic receptor subtypes, right? So what's really great is that the odd numbers are going to be M1, 3, and 5 are going to be excitatory, right? And that's going to mediate calcium increases in GQ pathway, right? So again, these are all going to have a GQ pathway. Remember, GQ is our phospholipase C, IP3, and DAG pathway, which is going to increase calcium, right? That's going to be our M1, 3, and 5. So again, remember that your odd numbers are going to be your excitatory muscarinic receptors, right? That's what's indicated in the box. Remember, M3 is the one that they really want you to note because that's the one that's most populated in the periphery. And then you also have basically your muscarinic receptor that are going to be inhibitory, right? So M2 and M4. Right, so again, M1, M3, and M5 are going to be excitatory GQ pathway. M2 and M4 are going to be inhibitory via the GI pathway, right? Which makes sense because remember, GI is going to be our inhibitory for GPCRs, right? So that's just going to be inhibit adenylate cyclase, inhibit cyclic AMP, eventually inhibit PKA and decrease phosphorylation, right? So those are going to be basically inhibitory, M2 and M4, right? So again, so what's interesting about these muscarinic receptors is that so M2 basically you typically find that in the heart, right? So when it's decreasing heart rate, basically not so much contractility, I would say more so decreasing heart rate. M2 is going to be the one that's going to be responsible for that, right? So I would associate M2 more so with the heart, right? M4 is not I would say not as high yield as M2, but it typically can modulate neuroexcitability. But again, I would associate M2 with the heart and decreasing heart rate, right? I wanted to touch upon this on the last slide, but I would also associate M3 with GI tract and bladder, right? So again, M3, it's more so like with increasing GI secretions, right? So or releasing basically the urine from the bladder, right? So that's going to be M3 versus M2, right? So M3 in the GI, M2 is more so heart, right? And obviously parasympathetic responses. Okay.
I have a quick question, Justin.
Yeah, of course.
What lecture was that GQ thing in again? GQ and like GS and stuff. What lecture was that?
I think that was Dr. D'Agostino's, I think third, but it was definitely in his series of lectures.
Okay, thanks.
Yeah, no problem. My bad. Okay, so now we have adrenergic. Remember, adrenergic is going to be only in the sympathetic nervous system, right? So again, just associate adrenergic, which makes sense, right? Like adrenaline, right? That's going to be a sympathetic response, right? Which is why we typically associate that with sympathetic nervous system. Okay, so another point that we touched upon, remember that our sympathetic nervous system is broader, right? Like we said, remember with epinephrine, it's a very broad effect. Right, while peripheral nervous system is more so organ specific. Like, hey, I want to decrease the heart rate. I want to, or increase GI secretions. I want to bronchoconstrict. Right, that's going to be very organ specific. Here, what's really good is we've touched upon this a lot. Right, so this should kind of be a review. Right, tyrosine is very important for catecholamine synthesis, regardless if it's dopamine, norepinephrine, or epinephrine. Right, so tyrosine, remember the rate limiting step is tyrosine hydroxylase, right? That's really important. That's going to be the rate limiting step of the synthesis of catecholamines, right? You produce L-dopa eventually into dopamine. And remember that norepinephrine is the only neurotransmitter that is actually produced in the vesicle, right? So dopamine turns into norepinephrine once it's in the vesicle. Okay. So the most important thing about norepinephrine is that it's not basically terminated by degradation, but more so by reuptake, right? So you have norepinephrine that's more so taken back into the presynaptic terminal, broken down by MAOA, which is monoamine oxidase, and it's basically going to turn it back into tyrosine eventually, right? So that's what's more so important for termination of action for norepinephrine is reuptake, not degradation, right? You could basically probably degrade norepinephrine, but it's more so associated with reuptaking it for MAOA, regardless if it's the presynaptic terminal or if it's even the postsynaptic terminal, which would be this number 2, because you can see norepinephrine goes into the postsynaptic terminal and then COMT breaks it down. Right, so again, that's very important to note because you can know it can be recycled into vesicles and it can be metabolized by MAOA. Right, that's more so associated with norepinephrine. Again, you have your auto and your heteroreceptors here. Again, remember your heteroreceptors are the ones that give feedback, right? So you'll learn a lot about that in PharM, but again, these are like typically receptors that can tell the presynaptic cell, hey, release more, or hey, stop releasing, right? So these are kind of like our feedback mechanisms at the synaptic cleft level. Okay, so which muscarinic receptors are excitatory and which protein pathway do they typically use, right? So I'll give you a few seconds on this one. Any questions while we wait?
I just looked up those pathways and it was in the postsynaptic signaling one slide. I think like 17 was the big like summary one, in case you guys were wondering.
Okay, perfect, thanks. Yeah, those GS, GQ, GI, I made sure to like put like a big star on that slide because Dr. D'Agostino likes it, and it's obviously going to be important for when we talk about these receptors, right? Because muscarinic, remember, are GPCRs, so they go through that G protein pathway. Okay, so seems like everybody's on the same page. Which muscarinic receptors are excitatory and which G protein pathway do they typically use? That's our odd numbers, right? So that's our M1, 3, and 5, and that's going to be through the GQ pathway, right? So they're typically going to use GQ. Remember, our GQ is our phospholipase C pathway with IP3 and DAG. The GS is actually our pathway for adenylate cyclase, cyclic AMP, etc. Okay, so adrenergic receptors, these are also going to be GPCRs, right? So these are going to be, you have your alphas and your betas, right? So the beta receptors are actually very similar genetically. The alpha receptors are only similar based on pharmacological indications. So these receptors basically obviously don't have channels associated with them. They're all GPCRs. All right, so basically I made this summary slide to talk about the alpha versus the beta adrenergic receptors. So this, I would say, is going to be the high yield stuff that you want to know regarding the differences between alpha and beta, right? So there's a lot that goes on in the next upcoming slides, but again, this is obviously, I would say, the more high yield stuff. So we have alpha versus beta. So the first one we'll talk about, basically, remember, they're all GPCRs. So you have alpha 1. So typically alpha 1 is going to be in your heart and your vascular smooth muscle, right? So if alpha 1 is going to be through our GQ pathway, right? So remember, I wrote the pathway down. That's our phospholipase C pathway, right? Its effects are going to be vasoconstriction, right? Because again, vasoconstriction and increasing contractile force, because that's going to be a GQ pathway, right? Because it's activating these things, right? It's going to be increasing contractile force because it's found in the heart. It's also found in the smooth muscle, right? That's our alpha 1 receptor. So if you have an alpha 1 agonist, right, it will cause vasoconstriction and increased contractile force, or an antagonist would do the opposite effects, right? So when we go over these, you really want to know the effects, and you also want to know what an antagonist would do, which would be the opposing effects, right? So that would be our alpha 1. Alpha 2, I would say just more so associate that with the pancreatic islets, right? You have alpha 2 in other locations, of course, but it's more so, I would say, just associating now with the pancreatic islets. And what's interesting about alpha 2 is that alpha 2 is actually inhibitory, right? It's actually the only one that I'll talk about that's inhibitory, and it actually decreases insulin secretion and decreases neurotransmitter release, right? So alpha 2 is like the odd one out, kind of. So it's the one that actually decreases sympathetic response, right? So it's like, All these other ones are going to increase sympathetic response because remember, they're like adrenergic receptors, right? So typically they're going to increase sympathetic response, but alpha 2 does not. Alpha 2 actually inhibits sympathetic stimulation, right? So alpha 2 is the complete opposite, right? So it's actually going to decrease insulin and decrease neurotransmitter release, right? And this is our GI pathway. Beta receptors, what's really good about the beta receptors is that they're all GS, right? So you could just be like, all right, beta receptors GS, right? So they're going to be GS pathway. Beta 1, the first thing, beta 1 is actually very popular. It's very well-known adrenergic receptor, right? The first thing you should think about beta 1 is the heart, right? So, for example, there's a lot of pharmacological drugs that are used as beta 1 agonist or a beta 1 antagonist, right? So if you're a beta 1 agonist, it's going to increase heart rate and contractility, right? Also increase renin release. But typically, beta 1, just think heart, right? So beta 1, heart. Beta 2, I think, okay, so beta 1, heart. Beta 2 is going to be lungs, right? So typically I would associate beta 2 with the lungs because there's a lot of beta 2 receptors in the lungs, right? So if you want to, I don't know if this helps, I just thought about this now, but beta 1, you have 1 heart, right? So that would help with the heart. Beta 2, you have 2 lungs, right? So maybe that helps. But again, beta 2 is going to be for the lungs and smooth muscle, right? So again, remember, since it's GS, it's going to be during a sympathetic response, right? So you're running away from something, and you want to bronchodilate, right? Because remember, beta 2 is highly expressed in the lungs, right? Highly expressed in the lungs. So for example, so with asthmatic patients, right? You would never want to give an asthmatic patient a beta 2 antagonist, right? Because that would cause bronchoconstriction. You don't want to cause bronchoconstriction in asthmatic patients, right? So for example, that's how these things are very clinically relevant, right? Because there's a lot of drugs that are used in regards to these alpha and beta receptors. And then last one, I would say beta 3 is on the lower yield side, right? Alpha 1, 2 and beta 1, 2 are very high yield. Beta 3 is on the lower yield side. Obviously good to know, of course, which is why I put it on here. It's more so associated with the adipose tissue, increases lipolysis and nitric oxide release, right? This is an important note that he actually talked about on the slide, is that cyclic AMP is going to increase heart rate and contraction, which makes sense, right? Because you can see in the beta 1, it goes through the GS pathway, right? So you're increasing cyclic AMP, which thus increases heart rate and contraction, right? But it actually dilates basically the blood vessels, right? So it's also going to cause dilation of the blood vessels. So you can see increased cyclic AMP here causes dilation of the skeletal blood vessels, right? And causes, for example, smooth muscles to relax. Right, so that's why, for example, lungs, blood vessels, uterus have beta 2 receptors, because if cyclic AMP goes up, that causes relaxation of the muscle, right? So again, I would associate these things with their prospective locations, right? So I feel like this is a really good summary slide to just hone in on your receptors, specifically adrenergic. Does anybody have any questions regarding this slide? I know it's a lot, obviously, there's a lot of receptors. Okay.
Maybe this is a dumb question, but those are like the high yield organs for those receptors, but they're on other organs also?
Yes.
Okay, thank you.
Yeah. So, question?
Yeah. For the beta 3, kind of a dumb question too. I think you had said before with that NO, that's nitric oxide, right? You're not saying like no release, you're saying like
sorry, yeah, that's nitric oxide, yep.
Okay, thanks, man.
Yeah, my bad. Yeah, nitric oxide. Okay, so that's going to be the high yield. Obviously, these receptors are mixed between some other structures, but these are the high yield locations.
Can you clarify?
Is there a question?
Sorry.
No, you're good.
What does AC stand for again? Sorry.
Oh, adenylate cyclase.
Thank you.
Yep. Okay, yeah. I'm glad. I hope breaking it down like this helps. Yeah. Yeah, so because I know it's a lot of receptors, right? So I just put it on one lecture and tried to really hone in on it with colors, and I like things in bullet points and stuff like that. That's how I study, so hopefully it helps you all as well. Okay, that's exactly. So basically, what we covered in these next two slides is what we just covered in the receptors, right? Because it's a lot. It's a lot looking at like this here, right? Which is why I broke it down in the last slide, right? So you see our alpha 1, vascular smooth muscle, and more so heart. Remember, increased vasoconstriction, contractile force. Alpha 2, remember, that's the one that is the opposite, right? Alpha 2, I'm always like, okay, that's triggering me. That's like the one that is going to be opposite to sympathetic nervous system, right? It's actually going to be GI and decrease insulin secretion. Then again, we have our last ones, which are betas. Our betas are all going to be GS, which is great, which is our adenylate cyclase pathway. Remember, the first thing you should think about with beta 1, remember beta 1, 1 heart, beta 2, 2 lungs. So again, beta 2, you're typically going to have those in the lungs. You'll have some in the GI tract as well. And then you have beta 3 on the lower yield side, but of course, nice to know, adipose tissue. So this is obviously a very high yield slide he wants you to know. So I would note what is in yellow, right? The yellow is going to be the most important part that is going to be testable material, right? He doesn't even highlight anything in beta 3, which is why I said, oh, it's kind of low yield, but still nice to know, right? So again, I would refer to this slide as well. Okay, any questions before we end off with the nested loops?
When they tested on receptors, what kind of questions? Was it more like matching them correctly, or a clinical question, or a drug agonist question? Like, how did they ask?
Yeah, so I could say it would be a little bit of all, basically. So again, agonist and antagonist, I would say would be like the simpler questions they can ask, right? But again, they can also ask patient questions, right? So regarding, so one of the questions I have on the practice quiz today, you'll see is patient-centered one. So you'll see that one. But basically, you can ask any questions regarding these. This is why I want you guys to like understand the key concepts, which is like the main location, its main effect, and if it's GS, GQ, GI. Right? Because if you know that, then you should be good regarding agonist, antagonist. What would happen to a patient if they have norepinephrine, adrenaline, or epinephrine, etc. Right? Does that make sense?
Yes, thank you.
Yeah, no problem. Yeah. All right. So we have our nested loops, right? So nested loops just means basically you want to preserve homeostasis, right? So if the brain breaks down, you have spinal cord responses, which can obviously assist. If the spinal cord is damaged, the ganglion can assist. If the ganglion are damaged, the synapse can assist, and eventually to the organ. That's basically this nested loop, right? That can basically help out with the functions of the things below, basically. So our outermost loop, we have our brain, right? Our cell bodies originate in the brainstem and are regulated by centers much higher up, right? So we'll talk about this later at the end of this lecture, but your heart rate basically is regulated by lots of things, right? We know the brainstem can help regulate the heart, right? But even your cortex, right? So think about when you're like if, for example, in classical conditioning, for example, if you train something to be scared of a buzzer, right? Or some certain buzzer or clicker, let's just say a clicker, right? And so, like a rat sees that clicker without the buzzer, they can already have an increased heart rate because they're already associating that clicker with, for example, a shock, right? Because you're already having that sympathetic response, meaning that that's cortical activation, right? Because your cortex is already telling you, hey, I need to be scared because this thing is about to shock me. Right, so that basically says that your cortex, which is higher centers, is actually regulating your heart rate as well, right? Because it can increase your heart rate because it's telling you you're scared before something even happens, right? So that's why it shows you that higher brain centers can regulate the heart as well as typically the brainstem. No need to know these things, right? We'll talk about the nucleus of the solitary tract a little bit, but the other stuff, like, don't even memorize that. You don't need to know that. Okay. So this is basically a feedback loop, basically, and this is kind of like completes the nested loop, right? So afferent neurons that detect pressure. So for example, our carotid sinus or arch of aorta is basically going to detect changes in pressure and send it to our, for example, our brainstem, right? And it's going to affect that, right? So for example, let's say you're scared and then your blood pressure goes up, your heart rate goes up, and eventually you're not scared anymore and you want to decrease your heart rate. For example, your carotid sinus or something like that, or your baroreceptors will be like, hey, we need to decrease this blood pressure to get it back to normal, and sends it back to the brainstem. So it's basically this entire loop that's going back and forth between efferent, so sending your motor signals, and your afferent, giving you feedback as to what you should change, if you should increase this or decrease that. Right, so that's basically what's going on here, right? This little thing, we'll talk about that in the upcoming slides. Yeah, so I won't touch on that right now. Okay, and that was the brain, right? Oh, that's going to be right here. So you can see. So the only thing I would note if you were to know anything would be sensory input typically goes to the nucleus of the solitary tract, eventually goes to the hypothalamus, and eventually goes to the cortex, right? So and then the cortex is like, hey, what should I do with this information? Should I do this, or that, et cetera, right? So that's our sensory afferent input, right? And like he says in lecture, he's like, oh, you have a monosynaptic, which means just like a one axon to one axon from the nucleus of the solitary tract to the hypothalamus, right? So if we go to the previous slide, I think, yeah, this slide is easier to see. So you can see in the nucleus of solitary tract, it goes straight to the hypothalamus in one synapse, or basically one fiber, right? That's why it's monosynaptic, right? But with the hypothalamus, from the hypothalamus to the cortex, it's polysynaptic because you obviously have like lots of synapses everywhere in the cortex, right? So that's what he highlighted in the slide. So I would say if you note this should be suffice in regards to this slide. So here, I would just note what is in red, right? Because he only specifically talks about what is in red. So that's basically amygdala cortex. So again, this is the association between autonomic nervous system and behavior. So again, this is the example that we just talked about, right? So heart rate can go up via classical conditioning, right? So rat is afraid of clicker because clicker causes shocking. Rat sees clicker, rat's heart rate shoots through the roof, right? So that's basically evidence saying that your cortex is going to basically, or higher centers, even like the amygdala, is going to assist in regulating heart function, right? Because it's going to increase your heart rate before something even happens. Right, so that shows that there's basically lots of connections basically between this emotion, these responses, et cetera, right? So you have this feedback loop basically. And what's happening is, so think about your baroreceptors. So if you have an increased mean arterial pressure, or like, let's just say an increased blood pressure, right? Your baroreceptors are like, hey, we need to bring this blood pressure down, right? It sends it to the vasomotor center. The vasomotor center is like, hey, we need to activate parasympathetic output because we need to decrease this high blood pressure, right? It decreases heart rate, decreases cardiac output, and then eventually decreases mean arterial pressure, right? It has less of an effect on contractile force. Parasympathetic is more so only associated with heart rate, right? Because typically, like your heart rate, if you didn't have any parasympathetic response to your heart rate, our resting heart rate would be much, much faster, right? Because the heart is typically has a standard heart rate is way faster if it wasn't because of this parasympathetic output that we had that was constantly affecting our heart, right? All right. And then we have the opposite pathway, right? So if you have a low blood pressure, baroreceptors are like, hey, we need to increase sympathetic output, increase heart rate, contractile force, venous tone, etc. And then you increase your mean arterial pressure, right? So with orthostatic hypotension, what's happening is the baroreceptors are underactivated, right? So for example, if you're sitting down and then you stand up quickly, your blood pressure raises quickly so that you can get the blood to the rest of your muscles and stuff like that, right? Because you're standing up and changing your position rapidly, right? In patients that have orthostatic hypotension, the baroreceptors are not responding to that change of standing up. And what happens is that you have a low blood pressure when you stand up, and it can actually cause you to pass out or fall over, right? So basically, your baroreceptors are very underactivated in orthostatic hypotension, right? They're like not as sensitive. So here's a little question. So a beta-1 antagonist would mainly lead to which of the following actions, right? So beta-1 antagonist. Any questions while we wait? Okay, so a beta 1 antagonist, right? So when I think beta 1, I'm always thinking, remember, beta 1, heart, so that's going to be for the heart, right? So it actually increases heart rate, right, and contractility. If you are an antagonist, that's going to lead to decreased heart rate and decreased heart contractility, which is going to be C, decreased heart contractility, right? So very good job on that one. I know these receptors are a lot to know right now, but again, little by little, we'll be able to get them down. Okay, so this is like our James Lange theory, right? Do you run because you're afraid, or are you afraid because you run, right? This is basically saying that these higher centers, example like amygdala, can have an impact on your autonomic nervous system and vice versa, right? So he talks a little bit about cognitive labeling. Remember, that's basically like, so he basically did a study on how, like, if you drink alcohol, right, that can lead to increased heart rate and flushing, right? And then your body's like, hey, my heart rate is high, I have flushing, I should be scared or angry, right? And that's basically what cognitive labeling is, because you labeled that physiological response an emotion, right? When it was simply just a physiological response. And that's basically saying that physiological response drives basically your emotion. And that's what James Lange theory is, right? That's basically for MCAS studying too, right? So that James Lange theory. Okay, so in this study, it's just simply showing that rats who are spontaneously hypertensive, they're just going to have typically a higher mean arterial pressure, which makes sense, right? If you're hypertensive, you'll probably have a higher base at which your mean arterial pressure is. If you were to put these hypertensive rats in a dirty cage, which is like in a stress environment for them, they have actually a huge increase in their mean arterial pressure, much greater than normal tensive rats. So you can see right here, much greater than these normal tensive rats, right? And that just means basically that higher centers in the brain are interacting with lower levels of the brain, and this affects the autonomic nervous system, right? Because stress basically is going to be induced by like your thoughts in your higher brain centers, and that's going to affect basically heart rate and mean arterial pressure, which would be regulated by lower brain centers, right? So this is just showing the connection between those two centers. So CFOS just shows measures neuronal activity, right? So rats that are hypertensive, right, have more CFOS activity because, again, remember, that's going to lead to the stressful environments, right? So when these hypertensive rats are in that dirty cage, they're going to have more CFOS activity than normal tensive rats, right? So a larger increase, like we talked about in the previous slide. So again, so what's interesting with MS and Parkinson's disease is that, so with Parkinson's disease, the earliest sign is actually dysautonomia, right? So actually effects in blood pressure as well. Those are actually going to be the earliest signs of Parkinson's disease before even the movement disorders, or even if it can lead to dementia disorders as well, right? So that's just basically saying that these diseases can impact at the autonomic nervous system. MS is more so in the higher centers of the brain, right? So more so in the vasomotor center. Parkinson's disease is more so at the level of sympathetic output, right? So basically control. All right, so here you have Parkinson's disease versus Lewy body, dementia Lewy bodies, right? So Lewy bodies more so associated with dementia happening first, and then movement disorders. Parkinson's disease can be like movement solely. Parkinson's disease can also lead to dementia as well, so that can also occur after, but Lewy body is more so associated with dementia. So you have control, right? So this is just testing for tyrosine hydroxylase. Remember that tyrosine hydroxylase is the rate limiting step for catecholamines like dopamine, right? So dopamine, remember, is affected in Parkinson's disease. And you see basic control. In a control, you have a lot of tyrosine hydroxylase, meaning a lot of dopamine, right? In the Parkinson's disease, almost none. And in dementia Lewy body, you basically have none as well, right? So this is just showing basically the different levels of how Parkinson's disease basically has very low levels of dopamine, for example, or tyrosine hydroxylase, even enzymes to form this dopamine, right? Because they're dying off. So this is our orthostatic basic kind of like orthostatic hypotension, right? Because you need to increase your blood pressure when you stand up. But what's happening here is that in controls, when you stand up, your norepinephrine increases because you want to increase the blood pressure so that blood can go throughout the body quickly and evenly. But in Parkinson's disease patients or dementia Lewy bodies, this increase in norepinephrine is not as high when patients stand up, which can lead to possibly orthostatic hypotension, right? And maybe even passing out or falling over, right? When you stand up too quickly. Then we have our spinal cord, which is going to be the next one. So what's interesting about the spinal cord is that, this is basically just saying if the brain was to be affected, the spinal cord would take over, right? So in this scenario, basically what's happening is that, let's say you have a spinal cord injury, but the reflex is intact, right? So spinal cord injury and the reflex is intact. What's happening is, let's just say your bladder basically is starting to fill up. It's starting to be distended, right? Starting to fill up, right? And you have basically a spinal cord injury, but the spinal cord reflex is still intact, right? What's going to happen is that the spinal cord reflex is going to cause you to void whenever, basically uncontrollably or involuntarily, you will not be able to control it, because what's going to happen is that if the connection between the spinal cord, for example, and the brain is not functioning for the bladder, let's just say, for example, what's going to happen is that the bladder, when it's distended, for example, is going to send a signal to the spinal cord saying, hey, we need to void, right? The spinal cord is going to be like, it's supposed to send it to the brain, but there's no connection there. The spinal cord is going to say, okay, we need to send this back to the bladder and say, okay, void. What's good is that, yes, you're going to void, right? But you're going to void involuntarily, so you will not be able to control when you void, right? Which would be enuresis, right? So you won't be able to control when you void. Not good, but better than hydronephrosis, right? And hydronephrosis would happen with MS patients, right? So MS patients, for example, there's no sensory basically happening. So what that means is that, so for example, the brain has no idea when the bladder is full because the sensory neurons from the bladder are not working, right? So if the sensory neurons that are supposed to send signals back basically are not working, then the brain has no idea when the bladder is full and has no idea when we need to urinate, right? So what's going to happen is that that's going to lead to hydronephrosis, because basically urine is going to go back into the kidneys because it's filling up so high, and then can lead to major kidney damage, right? Because the brain has no idea when the basically bladder is fully distended, right? So again, those are the key differences between MS. MS can lead to like that hydronephrosis or bladder dysfunction. The spinal cord injury would lead to enuresis, which would be involuntary control avoiding, but at least you're avoiding so that you don't basically destroy your kidneys, right? So those are the key differences between spinal cord injury and MS, right? Any questions? Is everybody good with that? Okay. All right, so then we have our ganglion, right? Ganglion, so basically if the spinal cord is injured, what's going to happen is that you actually have ganglion in your heart that actually can regulate heart rate and contractility, right? So the heart is very good at regulating, even by its own, basically, its own heart rate and contractility, right? So if these ganglion are affected, it can lead to congestive heart failure or even arrhythmias, right? And this is just showing that you have feedback at the ganglionic level. So you can see this receptor right here is giving feedback to this ganglion, right? So you have feedback even at the ganglionic level. And then lastly, synapse, right? So don't memorize these. You don't need to memorize them, he said, not for the exam, but it's just cool to know that we even have, for example, feedback. This is what I talked about with the heteroreceptors, right? These heteroreceptors say, hey, we should increase release or decrease release. This is feedback basically even at the synaptic level, showing, hey, if you have, for example, acetylcholine is released at an adrenergic synapse, that means to decrease the activity, right? Because it's telling you, hey, too much norepinephrine is being released, we need to decrease that, right? Or vice versa. And then here, this is just showing that angiotensin 2 can lead to direct effects on the synapse, right? Can lead to increased response because remember, angiotensin 2 is very highly associated with sympathetic responses, right? And it can lead to a direct effects at the synapse. And that is it. Anybody have any questions?
Hey Justin, I just wanted to like check in, I guess, on the whole like amygdala CFOS thing. So sorry, let me get back to what I was thinking with that.
No, you can.
CFOS is showing the immunoreactivity of the amygdala. So that's showing like the physiological response, whereas the level of the activity of the rat, like the rat is moving around more than the non-hypertensive rats. And so that's showing that emotional response to that physical stimulus, correct?
Can you restate your question? I'm not too sure what you're asking.
Yeah. So because they were trying to show like the link between like the cognitive labeling being like the link between your emotion and your physiological response. So the CFOS, the response to the CFOS, which is like essentially marking the activity in the amygdala, that's talking about the physiological response. Like that increased activity in the amygdala is the physiological response, whereas like then because those rats actually had more activity, that's what's like measuring the emotional response.
So the emotional response is more so associated with the amygdala, which is typically like higher brain center. When we think lower, we think more so brainstem, right? So brainstem regulating like primitive functions like heart rate, lungs, etc. So basically, what they're showing is that the connection basically between the amygdala, which is more so associated with emotion, right? So being scared, and then showing how that amygdala emotion can affect the brainstem, which brainstem just solely can, for example, regulate heart rate. How that emotion from the amygdala can tell the brainstem, which is lower neurons, to say, hey, we need to increase heart rate because we're scared. Does that make sense?
Okay, so for because the whole James Lange theory was like, I thought he was saying that it was more about the physiological response affecting your emotion. So would the physiological, like the measurement for the physiological response would be the heart rate then, or like the blood pressure, I guess. And then yeah. Right?
Okay. Yeah. So yeah, James Lange is physiological response and then your emotion, right? So basically that was with the cognitive labeling. So cognitive labeling with alcohol, increasing heart rate, and then you're like, okay, I should be angry because my heart rate is up. That's James Lange, right? There's obviously other theories as well, obviously talking about those things, but the main point is just knowing that there's a connection between these higher centers like the amygdala versus the lower centers like the brainstem in regulating heart rate.
Gotcha. Okay, so then the amygdala would be like that emotional response because that's the higher center that regulates emotion.
And fear and anger, stuff like that. Yep.
Okay. And then if anyone else has another question, I do have one more quick question, but I don't know if anyone else wants to ask anything first. Okay. Okay, you can go.
I was curious about the spinal reflexes referring to the afferent and efferent neurons. Like for the spinal cord injury, but the spinal reflex is still intact. Like, are the afferent and efferent neurons still severed, or are they still working?
They're still working for the spinal cord because you send the signal to the spinal cord, right? But what's not intact is that connection from the spinal cord to the brain, right? So then your brain has obviously no idea when you're basically distended, but what's happening is that your spinal cord says, hey, I'm receiving a signal basically from the bladder saying, hey, it's distended, we need to release, we need to void. So you're not going to have any control of that, but you're going to void to avoid hydronephrosis.
All right, thank you.
Yeah, no problem.
I had a question about one of the older lectures. So I was pretty with the whole like GABA and IPSP thing from way back when. I thought I had it, but now I'm not sure if I do.
Okay.
So like thinking about it in the context of your like practice question on your quiz, like that made sense because I understood that membrane potential was trying to move towards threshold potential. But in his example on his slide, I don't recall him actually saying that. So I was a little confused. And then I was also confused because if I'm correct, I think GABA releases chlorine. So I was confused as how that would in any way lead to depolarization of any kind.
Okay, so, wait, what was your first question again? I forgot.
Just like
what was the first
without
question?
him, without him blatantly stating membrane potential, how do we
Yes.
know that it's moving towards threshold?
Yeah, so membrane potential, remember, is always following equilibrium potential, not specifically threshold, right? It's not following threshold, it's following equilibrium potential of the ion most permeable, right? So if it's, for example, sodium, we know sodium's equilibrium potential is positive 67, right? If the membrane potential is negative 70, it wants to become positive 67, and thus will have to cross the threshold potential, which is typically negative 50, right? But let's say you have that would be obviously an EPSP. Let's say you have an ion that has, let's just say, an equilibrium potential that's at negative 60, right? And your membrane potential is at negative 70, right? Negative 70 wants to be negative 60. So it's going to obviously depolarize to get to negative 60. But what's going to happen is it's not going to reach threshold, right? So if it doesn't reach the threshold potential, which is negative 50 typically, it's still going to be considered an IPSP, even though it is depolarizing. Does that make sense?
Yeah, especially when you think about like reversal potential and equilibrium potential being like more or less the same when it's a single ion.
Exactly, synonymous in that scenario.
Right. Okay, that clarifies it. Thank you, man. Appreciate it.
Yeah, no problem. Any other questions? Oh yeah, so central nervous system, sympathetic nervous system, GI relaxation, and sphincters. Okay, so basically, this is a question in the chat. You want for the sympathetic nervous system, right? So if you have obviously GI wall relaxation, that means you're not pushing out obviously stool, right? You don't want to do that, or urination, right? That's definitely not what you want to do when you're running away from an animal or something like that, right? Which is why the GI wall is relaxed. And the sphincters, for example, like the anal sphincter or like the sphincter regulating the bladder, right? That's going to be constricted because you don't want to urinate or defecate when you're running away from an animal, right? Does that make sense? Okay. Any other questions?
I'm just a little confused on like I guess the difference between the spinal reflex versus like the ganglionic reflex. Is it like because the spinal reflex it's sensory, so it goes back to the ganglia and then goes into the spinal cord and like has that interneuron where it basically like sends the is it the efferent out to have that motor function to like contract the bladder or whatever. Whereas like the ganglia would not have that interneuron, or it would have like another interneuron in the ganglia itself.
So are you talking about like the stretch reflex like we talked about before? Like
Yeah,
versus this reflex?
Yeah, I'm confused on like the difference between the two.
Yeah, so with the stretch reflex, for example, remember that's like when you, for example, you hit your like the thing with the patella, you hit your patellar tendon, it sends a signal, your quadriceps send a signal to your spinal cord and basically say, hey, we need to contract the quadriceps. But it also synapses at an interneuron, which goes to the hamstrings to say, hey, do not contract because we're trying to extend the leg. So basically, you have two signals basically working in opposite effects to ensure that you're extending the leg, right? In the bladder example, it's more so just back forth, basically, just bladder's distended, right? Sends it back and it says, okay, we need to void because we have no connection with the brain. It's not working, but we need to avoid hydronephrosis because the body knows that's basically something that shouldn't be happening. We'd rather void involuntarily than destroy our kidneys, right?
Yeah, and so then I guess I'm just confused with why the ganglia is different from that. Is it just like instead of going all the way to the spinal cord, it just goes to the like ganglia and then goes back to that?
So typically you can have the ganglia near the spinal cord, right? So basically it could just be a quick signal going back and forth. With the bladder one, remember the spinal cord reflex there, it's more so just bladder's distended, release. There's no like inhibition of like anything else or like opposite effects opposing each other like in a stretch reflex where we activate the quadriceps and inactivate the hamstrings, right? In this one, it's more so, all right, distended, all right, we need to void.
Okay, so it's just going from the ganglia, like from the sensation to the ganglia back to the sensation, like the area of sensation to have a motor response.
Yeah, because what happens is that in your bladder, like, so sensory neurons are sending to the spinal cord, right? And the spinal cord, it's technically going to be your dorsal root ganglion, right? Your dorsal root ganglion are typically what gets sensory neurons from your body, right? So it gets sensory, and then once it goes into the ganglion, it goes into your spinal cord, then eventually out the ventral side, basically the ventral spinal nerve, which is important for motor, and then that's eventually going to send it back to the bladder to distend, or to, void. Does that make a little bit more sense?
Okay, yeah, no, I get that, and then I guess just like I get confused on the ganglionic response, but maybe I'll look it up.
Okay. But if anything, just send me an email again, and I'll definitely get to that as well.
Okay, thank you.
Yeah, no problem.
Hey, Justin, I want to ask 2 clarification questions.
Yeah.
Okay, so for the parasympathetic nervous system, the preganglionic are super long and then are close to the organ. And then the postganglionic are short because they're at that same organ. Is that correct?
So they're short because the ganglion are close to the spinal cord. So since it goes from the spinal cord to those ganglion, which is right nearby, so they're short, right? And then the postganglionic are much longer because it has to go to that effector organ.
I'm asking for the parasympathetic one.
Oh, parasympathetic. Yeah, so the preganglionic, like you said, is longer, so it goes all the way almost to the organ, and then once it synapses at that ganglion, it has a very short postganglionic that basically induces the response.
And that's because it's already like at the organ that it's going to act on?
Yep, exactly, yeah, my bad, yeah.
Just wanted to make sure. And then my other question was for the feedback loop. So for the baroreceptor reflex, so if you have too high of a pressure, like I kind of got confused on the cardiac output and then the stroke volume, but I know it's gonna like increase the overall pressure. So can you just go over that real quick again, please?
So if you originally have a high blood pressure?
Right? Yeah.
Okay, so yeah, if it's originally high, right, it's going to basically go to those baroreceptors, and the baroreceptors are going to say, okay, we need to send this signal to the brain to decrease the blood pressure, right? And then it's going to basically cause parasympathetic output, which is more so associated with heart rate rather than contractility, right? So it's going to decrease heart rate. When you decrease heart rate, you're eventually going to be decreasing cardiac output and stroke volume, right? Because stroke volume, remember, stroke volume is cardiac output times heart rate, right? I think that's the right one. Yeah, and then basically then that's going to lead to decrease in blood pressure, right? Does that make sense, or anything else?
Yeah, that makes sense. So then the arrow is like thicker for the heart rate because it's going to act more on that versus the contractile force.
Yes.
And then the opposite of that, it's going to act on like all 3 systems. So those are just all evenly for like the when it's too low for the sympathetic. Okay.
Exactly.
Okay, that makes sense. Thank you so much.
Yeah, no problem. Anybody else?
Hey, Justin, maybe I'm like weird. We do have a quiz today, right? Because it's not posted yet.
You do, yeah. So I'll tell the TA to make sure to post that then.
Okay, thank you.
Yeah, no problem. Yeah.
I hope you have a great weekend. And yeah, I don't know if that's your dog in the background or someone else's.
Yeah, he's going nuts. I'll get him after. In a good way, obviously.
Oh, of course. They just want treats and then they'll be quiet.
Exactly, yeah. Okay. Anything else? Hold on. All right. Any more questions? Let's see. All right. If not, Sucharita, I can like help you with your question as well if you would like as well.
Yeah, hey Justin. Okay, so
Hi.
my question's like from I guess the second lecture and the reverse potential lecture.
Mm-hmm.
And so I just didn't understand like when it comes to Nernst equation and the Goldman's equation, I wasn't understanding like I guess I understand that, and also like when we're looking at the potassium and like the movement in and out of the cell, like I wasn't like comprehending that at all. So I just want to like revisit that and like have you explain that. And I think that ties into reversal potential. I'm not sure.
That's
so.
Okay. So which one's the first one you want me to like talk about?
Can we talk about the second lecture first, like the movement of the ions in and out of the cell?
Yeah. Let me pull it up.
Okay.
Okay. So you see my screen, right?
Yeah, I can see it.
Okay. So let me see. Which slide specifically, or do you want me to just generally just go off of a limb just to explain what's going in and out?
You can go off the limb and just start explaining, if that's okay.
Okay, yeah, gotcha. Okay, so basically, the first thing you have is equilibrium potential of ions, right? So the most ions that we talk about are like potassium, sodium, right, and chloride, right? And they all have their own prospective equilibrium potentials, which means like where they're most happy, right? Where they are basically happy with their influx and efflux, so that it's basically going to lead to no movement, right? And that's our Nernst equation. Our Nernst equation, remember, is our 58 over Z log of concentration out over concentration in, right? So if you were to do the Nernst equation for potassium, right? So the typical potassium concentrations, right? I think it's like 5 millimoles out and like, I think it's 145 millimoles inside the cell, right? So if you do 5 divided by 140, 145, times log times 58 over Z, that will get you the negative 84 millivolts, which is like our standard potassium equilibrium potential, right? That's normally what potassium is in the cell, right? You normally have very little outside of it and a lot more inside, right? So that's basically where the cell is most likely to be happy, right, for equilibrium potential, right? If you were to, let's just say, let me get to the slide. If you were to, where's the Nernst equation? Yep. If you were to change, for example, the outside concentration of potassium, right? So this outside concentration, right, that would lead to obviously changes in the equilibrium potential. If you change the inside concentration, it would also lead to changes, right? So for example, if you were to increase the outside concentration of potassium, right, that means you would take a log of a larger number, right? The log of a larger number is going to lead to a number that's more positive, right? So that's what's going to happen in regards to potassium, for example, right? So, and then we have the same thing for sodium, which is positive 67, and chloride, which is negative 33, right? And those are basically those happy numbers where the net flux is 0, right? So, for example, so the membrane potential we have is normally negative 70, right? And the net membrane potential always wants to follow the equilibrium potential of the ion most permeable. And what that means is, for example, potassium is typically the ion that's always permeable, is the one that's most permeable, right? Basically meaning going through the cell, right? So the membrane potential negative 70 wants to be negative 84, right? Because negative 70 is like, let me get to negative 84. And what is a way to get negative 70 to negative 84? Because to get from negative 70 to negative 84, I need to become more negative, right? And since potassium is the only one that is most permeable right now, how do we get the cell more negative with potassium, right? Does it go inside the cell or does it go outside the cell, right? And it has to go outside the cell, right? Because if potassium goes outside the cell, it makes the cell more negative because positive ions are leaving the cell, and that will get it closer to that negative 84 millivolts, right? So that's basically the Nernst equation in summation. Does that make sense?
Yeah, that makes sense. So it doesn't have to do with like permeability then. It just focuses on like one ion, correct?
Yes, just one ion. So that's why it doesn't deal with permeability, because it's just one ion. So only thing that affects it is concentration out or concentration inside.
Okay, well that makes more sense then.
Okay, and then what was the other questions you had?
So do you remember the slide that has like the 4 different, it's like the green circles and it had like different concentrations and he had like things like going in and out?
Yeah, this slide right here?
Yeah, that one. I just like wanted more clarification on this one. Like how
No, I got you. Yeah. So basically, so this first part right here, right? What's happening? So basically this first part, you have, remember the membrane potential is positive 50. Right, so the membrane potential always wants to get to the equilibrium potential of the ion most permeable.
Wait,
right.
Like I have a question. This might be down to like the membrane potential. Is that going to be given to us, or is that like something we define? Like, how are we going to know what the membrane potential is?
So if they don't give you the membrane potential, assume negative 70. So because that's typically the normal one, but if they do, then you have to use the one they give you.
Okay.
Does that make sense?
Yeah, that makes sense.
Okay. No, I'm just asking. Okay. So basically, the membrane potential is always going to want to follow the ion most permeable, right? Which is potassium. Obviously, is the only one here. So positive 50 wants to become negative 84, right? So again, if they don't give you an equilibrium potential, it's safe to assume here, obviously, that the equilibrium potential is going to be standard, which is negative 84 for potassium, positive 67 for sodium, or negative 33 for chloride. Right? Obviously, if you change these outside versus inside concentrations, they'll change. Right? So positive 50 wants to become negative 84. In order for that to happen, it has to become more negative. Right? To become more negative, positive ions have to leave the cell. Right? So that's why it's moving out of the cell. Here, the membrane potential. Oh, you have a question?
Well, no, yeah, that makes sense. I guess I want to ask, like, so would it be different if the concentrations inside and outside the cell were different? Like if it was like reversed, for example?
Yeah, that would make a huge difference, right? So if the concentrations were so different, right? So for example, potassium's normally 5 on the outside and 140 on the inside, and that gives you your negative 84 using the Nernst equation. If the concentrations were so different that it actually caused the Nernst equation to give you an answer that's like, let's just say positive 60, right? That would be actually more positive than positive 50. So then that would have to be influx of potassium, because to get from positive 50 to positive 60, potassium would have to influx, right? So it depends on how big the concentration changes, right? So depending on if it can actually change where the influx or efflux of the membrane potential goes. Right?
Okay.
Does that make sense or no? Or do you want me to explain it?
I think it makes sense. Yeah.
Are you sure?
It's just like I'm a visual person, so I kind of need to see it like happening, kind of thing, like an example. Okay. I could kind of hear it.
No, you're good. So I think there's a slide on that. Let me see. Hold on. Yeah, right here. So this slide is basically showing you exactly what we just talked about, right? So typically it's 5 on the outside and 140 on the inside millimoles. If you were to increase potassium, right, by 50 to 50, so from 5 to 50 on the outside, that means basically it would be, if you were to solve basically using calculator the Nernst equation, 58 divided by Z, which is 1, so 58 divided by 1 is 58, times the log of concentration out over concentration in, you went from 5 to 50, so that means the numerator got bigger, right? So the numerator got bigger. That's going to lead, the log of a bigger number is more positive, right? So that's typically going to cause the membrane potential to go from negative 70 to a more positive number, which is negative 40, right? I wouldn't be able to get negative 40 unless I obviously use the calculator, right? But I know it's going to be more positive.
Okay.
Does that make sense?
Yes.
Oh, excuse me. I meant negative 84 to negative 26. Same thing, but obviously.
Yeah. positive. Like I understand that now. Like
Does that make sense?
Yeah, it makes a lot more sense.
Okay.
Thanks.
Okay. Anything else, you sure?
And the Goldman's equation.
Mm-hmm. Yeah, this one. Yep. Or would you just like me to like run it down?
Yeah, just like kind of explain like what it's trying to find.
Yeah, so with Goldman, you want to find membrane potential, right? With Nernst's equation, you want to find equilibrium potential, right? Membrane potential is always going to follow the equilibrium potential, right? So membrane potential is always going to want to become equilibrium potential. So Goldman, membrane potential, Nernst, equilibrium potential, right? This is why permeability is so important for Goldman, because there's so many ions that are in play here, right? So here we have 3 ions that are in play, and if the permeability of one ion is much higher than the other, then that's going to change the membrane potential, right? So typically, like we said, the membrane potential is -70. Why is that? The reason why it's -70 is because the permeability of potassium is much greater than sodium and chloride, right? So that means that the membrane potential wants to be closer to -84, which is the equilibrium potential of potassium, right? It won't get there, obviously, because we still have sodium and chloride affecting it, but it will get close, which is why the membrane potential is -70, which is pretty close to -84, right? But if you change the permeabilities, for example, if you increase the permeability of sodium now and decrease everything else, now the membrane potential is going to want to move towards more positive numbers, closer towards positive 67, right? Does that make sense?
Yeah, that makes sense.
Okay.
I'm still trying to I feel like I still don't understand it, but at the same time it makes sense. I don't know if that makes sense either.
Yeah, no, I've had those moments. So what do you think is confusing you?
I feel like I'm still getting confused on just like the movement, like in and out, because I know like in that example with the 4 examples with the membrane potential and the potassium moving in and out, like the last example, it was like potassium was like moving in. So I guess could you explain that one real quick?
Oh, the one that potassium was moving in?
Yeah.
Yeah, I got you. Yeah. So right here. So remember, so if you tell yourself membrane potential follows the equilibrium potential of the ion most permeable, so -90 wants to become -84, right? Because that's the equilibrium potential of potassium. To go from -90 to -84, that means you have to be more positive, right? Because -90 to -84 is a positive movement. In order for the cell to become more positive, potassium has to influx because it's a positive ion to make the cell more positive.
Okay, that makes sense. So I guess like with the fact that there's like a lower concentration of potassium outside the cell, like kind of also affect how slow or fast it would go in. Like
that's a good point. Yeah, like you're saying, like how much potassium can go inside, which is why potassium almost never goes inside the cell, right? It doesn't need to, because typically potassium is always going to efflux. Right, that's almost normal. This is like an extreme case, right? Just in case, like this is just hypothetical. Does that make sense?
Yes.
Yeah. Okay.
Yeah. Okay. Because I think like
I think that makes more sense, eh?
Yeah, it makes more sense. Because the way I was thinking about it was like, okay, like, because I was thinking, I guess it's like concentration gradient where like it goes from like high to low, and so like there's such a low concentration outside the cell versus inside the cell. So I just like I wasn't like making the connection with like that plus the membrane potential, like trying to balance that out. Does that make sense?
Yeah, like wait, say it again. Wait.
So like so you know how the concentration gradient like it like diffusion or whatever, like it goes from high to low, like there's a whole high concentration on one side and then
yes.
Yeah, so like I guess like looking at this, I see it as like there's a high concentration on inside, a low concentration outside. So like in my brain, it's wanting to go outside to like kind of fix that. But then I know it's also trying to take into consideration like the fact that there's the membrane potential that has to like balance that out.
So it's because like you have to remember it's not only a chemical gradient, which is from high to low, right? It's also an electrical gradient, right? So you have 2 things that affect movement: chemical, which is high to low; electrical, which is our membrane potential. And you want to make both happy.
Yeah.
Right. But typically, you're always going to want to make membrane potential follow equilibrium potential.
Okay.
Right.
Okay, yeah, that makes more sense then. I get that.
Okay, good. Anything else?
On this lecture, no, but could we cover like the reversal potential stuff now?
Yeah, I got you. Okay, hold on. So, pretend. What lecture was that?
I'm not 100% sure.
I think is it 4 or 5? I don't. Let me see. Let me pull them up.
I think it's 5.
Yeah,
Yeah, it's 5.
It's 5.
Yes.
Okay. Five. Oh, wait, but which? I don't know. I'm getting confused with 4 and 5. Which slide on 5 is like towards the end?
Postsynaptic. Yeah, it's like towards the end, like right after he talks about like EPSP and IPSP, like reviews it and the meta.
Got it.
Yes.
Got it. Yes. Okay. Let me pull some. Oh, wrong one. Okay. This one, eh?
Yes, that's the one.
Okay. So what are you more so confused about?
The last part, the reversal of potential, like how
Yeah.
it's a depolarization. Could you just explain it again, please?
Yeah. No. Okay. So basically, the most important thing to know is that an EPSP is defined as the reversal potential being greater than the threshold potential, right? So for example, let's say here in this one over here, right? The reversal potential is 0, right? That means the membrane potential, let's say it's -60, always wants to follow the reversal potential of the ion most permeable. Right? Remember, reversal potential, equilibrium potential, interchangeable when you're talking about one ion. So it has to go past -40 in order to get to 0, right? So that means it's going to be an EPSP, right? Because it has the potential to pass this threshold to get to that reversal potential. In an IPSP, right, the reversal potential is, for example, let's just say -80. You want to go from -60 to -80, right? So you're going to become more negative, right? But you'll never be able to get to this threshold because the reversal potential here is not above the threshold, right? So no matter how many times you activate this GABA, it's always going to be an IPSP because the reversal potential is not above the threshold. So meaning this will never pass the threshold, right? In this example, let's say you haven't activated another GABA synapse, right? Let's say you have a membrane potential of, this is -60, right? So the membrane potential -60, but you have a reversal potential of -50, right? Remember, the membrane potential is always going to want to follow the equilibrium potential of the ion most permeable, or reversal potential. So this wants to go from -60 to -50, right? In order for that to happen, you want to depolarize, right? So depolarize, become more positive, right? But because the reversal potential, -50, is less than -40, that means this will never reach -40. And remember, if you never reach -40, you'll never fire an action potential, right? So then it has to be an IPSP, because remember, we want to get to that reversal potential. We don't want to, like, go above that, right? That's not the point, unless you're in an action potential, right? So once they get to -50, that's it. This will never, no matter how many times you activate this GABA synapse, it will never get to threshold because it only wants to get to -50, right? Does that make sense?
I think so. So is the reversal potential like a set number always, or is it going to change just depending on like the question that they ask?
So remember, reversal potential is synonymous to equilibrium potential when you're talking about one ion, right? So for example, if we're talking about potassium, we know it's -84, that's the reversal potential, right? Or equilibrium potential, right? It's the same thing. Or it can change depending on what ion, like, or concentrations, because remember, if concentration out or concentration in changes, it's going to change, right? Or if more ions play into factor. Does that make sense?
No.
Okay. So I got you. Okay. I like the honesty. Okay, so remember that the reversal potential, what it is basically when there's no net ion flux, right? So no ions going outside the cell, no ions going inside the cell, right? That's when it's happy. The reason why we use reversal potential is because we typically talk about that when there's multiple ions in play. So for example, when you have sodium and potassium, right, and they're both permeable. Let's say that now the membrane potential wants to be positive 67, but it also wants to be negative 84. So there's like a tugging, right? It's going in and out. So the reversal potential of that would be close to 0, right? Because that would be making sodium happy as well as potassium happy because you're in between both of them. That's when we use reversal potential, right? Because it's talking about multiple ions. When we're talking about one ion here, for example, like let's just say, I don't know, chloride or something, that's going to be basically the same thing as saying negative 33 for chloride is the reversal potential, also known as the equilibrium potential of chloride. Does that part make sense?
I'm gonna be so honest. No, I'm totally lost.
Okay, what are you confused about?
I am like I don't even okay like I guess how is it being balanced out with it being negative 64? Like I don't even know. I'm so lost.
No, you're good. Okay, what are you confused about specifically so that I can help you?
I guess can you try explaining that again, but just go a little slower so I can comprehend like.
Yeah, okay. So remember that reversal potential is the same thing as equilibrium potential for one ion. Right? Okay. Remember, equilibrium potential is that Nernst equation. Mm-hmm. That 58 divided by Z log of concentration out over concentration in. Right? So that's going to be, remember, standard for potassium is negative 84, for sodium positive 67, and then chloride negative 33. Right? If we would actually calculate them out. You get that, right?
Yes.
Okay. So that's reversal potential and equilibrium potential are interchangeable there. When we're talking about multiple ions like sodium and potassium both being permeable, for example, we know potassium reversal potential, also known as equilibrium potential, is negative 84. Remember that sodium is positive 67. So now you're going to have an efflux of, let's just say they're equally permeable. You're going to have an efflux of potassium as well as an influx of sodium, right? Efflux of potassium because you want to be more negative. You want to get to that negative 84. Influx of sodium because you want to get closer to that positive 67, become more positive. So the reversal potential is when there is no efflux of no more efflux of potassium and no more influx of sodium because the cell is happy now. So that reversal potential is roughly 0 for those two ions. Right? Because between negative 84 and positive 67, that middle number is close to 0. Okay. Right? So that's when the cell would be happy if you're permeable to both ions.
Yes, that makes sense.
Does that make sense? That's why they use reversal potential. Does that make sense?
Yes. Is the goal of reversal potential like to always have a net like just to have a net of 0 or?
Yes, to have no net flux basically. A net flux of 0, yeah.
Okay.
So that nothing's going out of the cell and nothing's going in, so that the cell is basically happy at where it's at.
Okay, yeah, I think it's just like confusing because there's so many like potentials, like
it is, yeah.
Really clever, like it's just I think they're all just getting confused in my head. But okay, that makes more sense. So does that? So could you explain like the C chart, like the C graph again, like one more time?
Yeah. So again, so now that we know about this reversal potential, right? Let's keep it simple and say we're talking about one ion, right? So same thing as equilibrium potential, right? Let's say the reversal potential is negative 50, like it's shown in this graph, right? Mm-hmm. The membrane potential is negative 60, right? Remember that negative 60, the membrane potential always wants to become the equilibrium potential, also known as reversal potential, right?
Mm-hmm.
Remember that?
Yes, I remember that.
Okay, so basically, negative 60 wants to become negative 50. So in order for that to happen, it has to be more positive, right? Because negative 60 to negative 50 is more positive, right? So that would have to cause a depolarization because depolarization means you're going more positive, right? But remember, like we said, reversal potential, the key is to get to that no net ion flux. So once it gets to negative 50, there's no reason for this to go to negative 40 because it's already happy at negative 50. There's no